All right, welcome everyone, and thank you so much for joining the 2021 Tilden Fungus Fair. I'm your host, Trent Pearce, and I'm a naturalist with the East Bay Regional Park District. I'm broadcasting today from Tilden Nature Area in Berkeley, California. I'm joined by my other host, Melissa Fowlks.
Good morning.
And we have our first guest panelist on the line, Christian Schwarz.
All right. Howdy, everybody. I'm Christian. I'm a research associate at the Norris Center for Natural History at UC Santa Cruz, and I'm broadcasting from Santa Cruz.
Great. Thanks for sharing, Christian. Hey, before we continue, folks, I want to hear from you using the question and answer option or the comments box in your Zoom webinar. I'd like to know where you're joining us from today. So go ahead and throw a comment in there. Let us know where you're watching this event from. Or if you're watching live on Facebook or YouTube, go ahead and throw a comment in there and tell us where you are in the world. All right, lots of locals. I'm seeing a lot of locals. Oh, Austin, Texas, groovy. San Francisco, Point Reyes. They have internet in Point Reyes? Wow. Oh, we've got our first international visitor, India. I was going to ask who's watching from the farthest distance away. New Hampshire. Wow, I hear it's very cold over there. Well, thanks so much, folks. I'm glad we can all get together and virtually share our mutual love of the fungi. Before we go any further, I'm going to give you a brief outline of today's event. There we go. So at 10 a.m., the event began. In just a few minutes, we're going to hear from Christian on the long-term mushroom monitoring lessons from a 3-year effort. And we're going to have a question and answer with Christian, and hopefully we'll squeeze in a short break. At 11:15, we're going to have a naturalist's guide to mushroom identification books. I've asked Christian to stick around and help me review and possibly heckle my extensive collection of mushroom field guides. Then we'll have another short break. At 11:45, we're going to hear from culinary wizard Morgan Evans. She's going to take us on a live cooking journey where she makes something fantastic with Matsutake. Then another break after that. 12:45, we're going to hear about the mushrooms of Lake Merritt with Damon Tighe, local naturalist extraordinaire. And another short break. And then at 1:45, I'm going to review the results of the Bay Area MycoBlitz, which was an event hosted on iNaturalist in the week leading up to this event. So in just a moment, I'll throw that link in the comments section. But at any point during today's event, you can slide on over to iNaturalist and you can view our virtual specimen tables. Since we can't have this event in person, and I know most of you like going to mushroom fairs so you can poke at rotting mushrooms on tables, we've done the next best thing. We've sent citizen scientists out into the field to virtually and digitally collect fungi, and we've aggregated them on this iNaturalist project page. So you can see everything that's been observed in the Bay Area for the past week on that page. So stand by for that link. Then at 2 o'clock, we're going to have a fungus trivia on Kahoot, and I promise it's quite challenging, and there will be prizes. So get excited about that. And then at 3 o'clock, the event will be over, and we'll call it a day. So that's the outline of today's event. Hopefully, we'll meet all of your mycophilic needs. And I'm going to drop that link into the comments box right now. Maybe. Here we go. So don't let this distract you too much, but here is that iNaturalist link. You can surf over there at any time and check out what we've been observing. All right. Well, I don't want to penalize folks for being on here early. I guess I can talk a little bit about our first guest panelist now, Christian. I'm going to tell a story about you, if that's okay. I promise it won't be too embarrassing. So I met Christian probably around a decade ago in Santa Cruz, California. I think we were at a random house party, and you can correct me on this story, Christian. We had a little exchange beforehand via some note swapping, but I think I actually met you in person at the house party. And I was pretty much immediately struck with this guy. He had a denim jacket on with a huge mushroom patch on the back, and he obviously knew a lot about natural history. So, you know, our mutual love for all things nature was kind of obvious. Little did I know he would go on to co-author the definitive guidebook to coastal California fungi and become a close friend.
It was really a mutualistic relationship because you knew a lot about parties, so I got to learn a lot from you.
Well, I'm glad I could in some way contribute. Christian's a naturalist, mycologist, and educator hailing from Santa Cruz, California. He studied ecology and evolutionary biology at UC Santa Cruz and now spends his time photographing, teaching about, collecting, and researching macrofungi. Let's be real, you dabble in the microfungi too. He's a research associate at the Norris Center for Natural History and the Santa Barbara Botanical Garden, and co-author of the wildly popular book, Mushrooms of the Redwood Coast, which we now abbreviate MORC. Please make virtually welcome Christian Schwarz.
Thanks, Trent.
Sure.
I just want to give a big thanks to both Trent and Melissa and everyone else involved in putting this on, because in a year like this, obviously, there's a lot of desire to sort of do the things that we used to do in normal years, have our mushroom gatherings in the fall and the winter. And I know people probably more than ever wish they were going to those events, but since we can't have them, it's really nice that people are making the effort to put these events on in whatever form we can have them. So big thanks to Trent and Melissa and all the other organizers. So today I'm going to be talking about this project that I've been working on for 3 years now in Santa Cruz. And I think I'm just going to start screen sharing now, if that's okay with you, Trent. Is that good?
Take it away.
Okay, cool. So I'll just, yeah, I'm not going to introduce it too much more. I'm just going to start the talk now. Oh, I got to get my screen sharing enabled. Trent or Melissa, am I allowed to share my screen now? I am now. Okay, here we go. All right, so this talk is called Long-Term Lessons, Perspectives from 3 Years of Macrofungi Monitoring. And this is really, it's my first attempt at sort of distilling some of the findings of this 3-year project. It's not even over. I'm still in the middle of year 3. It'll be done about April or May will be the end of the actual 3-year period. So let's get into it. All right. What you're going to see this abbreviation a few times throughout the course of this talk, so I just wanted to define it. LTMM doesn't actually stand for lettuce, tomatoes, mayonnaise, and mustard. Nope. It stands for long-term macrofungal monitoring. So that's what I mean when you see this abbreviation. And what do I mean by long-term? So obviously 3 years doesn't sound like a very long term. That's a relatively short period of time, especially for sort of natural history. 3 years is sort of the blink of an eye in an ecological sense. But what I mean here is surveying the same locations frequently and regularly over a multi-year or multi-decadal, if you can get away with it, time span, to try and get a sense of a location's progression through time in a slightly more rigorous way than we normally do. You know, as mushroomers, and this is especially true of me, we often sort of chase the rains around and go wherever the mushrooms are fruiting. But what that perspective often leaves us unaware of is what happens when it's not raining. What happens when conditions aren't perfect? How does the progression of a season look at a single location, regardless of what the weather conditions are? And so I wanted to get a better sense of this sort of ecology, do sort of disciplined monitoring of the same locations, where I actually also quantified my effort. And we'll talk more about what that means later, but it's really just a way of quantifying how much time you spent what area did you cover and how much time did you spend covering that area? Obviously, the longer you spend in a little patch of forest, the more mushrooms you'll find. So this just gives us sort of a divisor number. You can divide by the amount of time and area that you covered, and you can get sort of standardized ideas of how rare or how common a mushroom is within that area, given the effort spent. So this sounds a little bit complicated, but I really tried to make a protocol that was as simple as possible so that I wouldn't have a lot of trouble doing it and that other people could try and replicate it in their own backyards. My effort was focused in Santa Cruz, but my goal is for other people to do this kind of monitoring elsewhere in the state in different habitats and really across the country. Long-term monitoring efforts are sort of, I think, the most interesting way of getting data about how things are changing. You can sort of do haphazard efforts where you just go out and look at mushrooms, or you can make these slightly more structured, slightly more disciplined monitoring efforts and get some really interesting data about how biodiversity is changing around us over time. So my protocol that I came up with was that I was going to establish a set of transects, which just means trails, really, each of them 1 kilometer long, that I would walk out and back. So each transect takes me 2 kilometers of walking, but they happen in the same area. So 1 kilometer out, 1 kilometer back. I would record my start and end time so I could quantify the total amount of time spent. And I would record every species present that I could identify and count the abundance of each of those species. So this is what it looks like in the end, is that you have a list of species and then how many of each of them you saw within 1 kilometer, within 1 hour or an hour and a half, however long it takes you. That sounds simple, and it's intended to be simple, but there's actually quite a bit of statistical power you get when you do this repeatedly. So this is sort of a map of my observations of fungi in Santa Cruz County, at least in the past, what is this, 4 years now, 3 years now, the 3 years of this survey effort. And you can see that my observations are really clumped up around a few areas where my long-term monitoring transects are located. So I'm doing a lot less scattered mushroom hunting these days and a lot more focused, disciplined monitoring efforts of a few locations. So here's a visualization of the 10 transects that I set up. Each of them, remember, are 1 kilometer long. These are not all to the same scale. So some of these little maplets show 3 transects, some show 1 transect, some show 2 transects. But each one of these I would walk every 7 to 10 days. And people will often ask, okay, if you're walking the transect down a trail, how much time do you spend going off trail? Do you branch out at all? Which mushrooms do you count? The ones right next to you or the ones that are 10 feet away from the trail? And the way I explain it is that I made sort of an attempt to focus most of my effort within 1 meter on either side of the edge of the trail, with a lot less effort out up to 10 meters away. So if you take the red line in the middle of this effort curve, that's the trail itself. And so going 10 meters in one direction, 10 meters in the other direction, I spend very little time out at 10 meters, and most of my effort really is just trail side mushrooms, just the mushrooms that are within about 1 or 2 meters of the trail. And the reason I didn't make it super precise with quadrats and sort of randomized sampling is that I find those protocols in academic biology really onerous. They're really hard to replicate. And I felt like they were bad at capturing what was actually going on with mushroom diversity in any place at a given time. So I came up with this sort of looser, more intuitive, controlled method to sort of allow me to just do what I do, go out and record mushrooms and get a sense of a picture of what's happening at any given moment. So there's sort of the amount of effort that I spend in any given location or any distance from the trail. And then those are the dimensions. So you can imagine taking a map of one of these trails and putting that effort curve over any given spot on the trail, and you end up with a sort of curve of effort that looks like this. This actually looks sort of like some sort of intestinal diagram, but really it's just intending to show that there's a little bit of leeway on both sides of the trail that I count the mushrooms that I see. So when you do 10 transects every 7 to 10 days throughout the course of the rainy season for 3 years, the way I am almost completed doing now, you end up getting a lot of records. This is what my notebook looks like for 1 day. Actually, this ended up being only like about 1 hour and 45 minutes spent on 1 kilometer transect just a few days ago. And there was, I think I ended up with 90 species just on this 1 kilometer transect. So it was a really productive day. And so when you do this repeatedly for 3 years, I've ended up with something like 11,000 records of fruitings quantified by abundance. That took me about 500 hours to complete. And I will have ended up walking about 800 kilometers by the end of it. So this is sort of an intensive effort, and I really don't recommend doing it as intensively as I've done it. If I were to redo this all again, I would either reduce the length of my transects or reduce the number of them. I don't think anyone should feel like this is the best way to do this. If you're interested in doing a long-term monitoring project, I'd recommend choosing 1 or 2 transects near you that you don't have to spend quite so much time investing effort in. But the upshot of all this is that when you have 11,000 records, there's a lot of data to explore. So just to begin summarizing some of what I've found, species diversity in these transects or over these transects is really high. The least diverse transect in 2019, or in that season, 2018-2019, had 334 species on it. So that's the low end of my transect diversity. The high end was something like 435 species, but that was just for 1 year. At this point, with 2 more years of survey data, I think that particular transect is up above 450 species now. So these are extremely diverse, rich transects, mostly through redwood forest. The difference between the lowest and the highest diversity of the transects was about 100 species. So there is quite a big difference between that old growth redwood forest or mixed evergreen forest dominated by redwoods, and then the least diverse transect, which is the urbanized transect through the UC Santa Cruz campus that just sort of goes through the remnant interstitial forest between buildings. So here's what a big basin transect looks like. There's just oodles and oodles of mushrooms, lots and lots and lots of species. So the overall number of species that I've detected across all transects now greatly exceeds 550 species. I'm not exactly sure where we're at now. I have to tally up this year's totals. But there's a lot of mushrooms on just relatively small area of this county's land. 1 kilometer and then 3 meters on either side of it, or up to 10. Isn't very much percentage of this county's land area, and yet within that area you can find over 500 species of mushrooms, or macrofungi, I should say. So what have we found? Some distinct, I want to just cover these sort of 3 attractive, exciting categories: disjunct, rare, and undescribed species. So disjunct means those species that are common elsewhere, but they're rare here. They're sort of separate from their other populations. This may be the farthest south they occur. Rare species, that's obvious, species that are just rare everywhere. And then undescribed species, new species to science that have not yet been named. So here's an example of a disjunct species, Hayderia abiatus. This is a beautiful little ascomycete that grows on Douglas fir needles. It is fairly common further north and in the Sierra Nevada. It's not at all common on the coast, especially this far south. I think this is as far south as it's ever been recorded. That was at Big Basin. It's the only location that I had it on my transects. And it's just an example of one of these disjunct species that you can detect if you pay close attention. Lepiota rhodophylla. This is one that I saw just a few days ago. It was only known from one location anywhere in the world, up in San Mateo County, where it was originally described. But in the course of doing these long-term monitoring transects, I managed to detect it. And it has a very short window of fruiting, maybe just a week, where it's visible above ground. And that's one of the benefits of doing this 7 to 10 day sort of intense sampling regime, is that if things sort of come and go quickly, you're more likely to detect them than if you were just visiting casually whenever you felt like it. This sort of discipline allows you to intercept those species at the moment that they're fruiting. So this was a really good find that I was super excited to see. Mycinella, here's a species that is, the entire genus was not recorded from California, and I don't think there's many records anywhere in the United States, at least in publicly available databases, not under this name. But it was something that I found at Big Basin when I was doing my transects there. And I'll talk more about this later, but it survived the recent CZU fire. So I was really glad to see that this special mushroom that appears relatively infrequent throughout most of the state still is holding on to the one place we know it from in Santa Cruz County. And then there's these undescribed taxa, things like this trichosobe, which is actually present on multiple of my transects, but is not named. I'm hoping to work with Brian Perry at CSU East Bay, is that what it's called? Yeah, who does a lot of research on these white spored fungi, and hoping to get this one named if it's not already named. We think it's an undescribed taxon anyways. It's a poorly known genus around the world, yeah, it's a cool little mushroom with this fuzzy cap. I'll show you a close up right here, where it's got quite hairy caps when it's young. Grows out of wood. It's a white spored species. So it's sort of not even that exciting to find undescribed species in California, because if you're not finding undescribed mushrooms, you're just not trying hard enough. Undescribed mushrooms are kind of ubiquitous. There's way too many mushrooms that need names still in California. So it's not even that surprising to find them. What I think is actually more interesting is the disjunct species, those that we're finding at their southern range limits or away from their known habitats. So I just want to talk a little bit about lessons about how mushrooms behave in time and space that I've learned from this project. So the first one I want to talk about is fruit body persistence. And what I mean by that is a single fruit body, one mushroom. How long does that really last? We tend to think of mushrooms as being ephemeral. They come up and they go away. Maybe they last a week or two. Maybe some of them last a few months, like the big Leucopaxillus, if they're really resistant to decay. And then I'm going to talk about seasonal progression, which I think is a little bit more intuitive. But one thing that I accidentally learned about fruit body persistence came because I found this mushroom, Gymnopolis punctifolius, which is a pretty rare species in Santa Cruz. It's only known from a couple locations in old growth redwood forest. And the first year that I was doing this, I found it growing on a redwood log in Big Basin. And I thought, oh, wow, this is so cool. I rarely ever see this mushroom, but it was just one single fruit body, which was actually helpful because I was able to watch that exact same individual for the next few months. And I didn't at first realize that it was going to be so long lasting, but as I kept watching the fruit body, I became more and more interested in how long it was persisting there. And in the end, I have data showing that it was present for at least 105 days. So a single fruit body, a little gilled mushroom that you think of as something that comes up and rots away really quickly, lasted for 105 days, which totally challenged my perceptions of how long mushrooms last. At least some of them, it appears, can last much longer than I had originally realized. And it makes sense for this species. There's some strange chemistry going on in it. It does seem to be rot resistant in a way that suggests that it has chemicals in it that support this durability, this duration through time. But it's the kind of thing that I would never have gotten the data about if I hadn't been doing repeated and persistent surveying of the same location. So that was sort of an accidental but really valuable find for my sense of the natural history and ecology of mushrooms. Diversity through the season. So this is the seasonal progression of mushroom diversity and abundance. And what you see if you plot the number of species on the vertical axis, the y-axis, and then a progression through a season from the start of the rainy season in October to the end of the rainy season, approximately in April, you once again get this common pattern that repeats through a lot of nature, which is the bell curve. The middle of the season, when it's rainiest and wettest, is peak mushroom season. That's when you get the greatest abundance. So the number of species on each transect was highest in the middle of winter, approximately December and January, and then it declined again into April. So this is an expected pattern. This is sort of what we expect to see. Now, changes in this graph, this only shows 1 year worth, one season's worth of data. Changes in the shape of this graph, whether it gets a narrower bell or a wider bell or two peaks, these are all potential changes that we might expect to see as climate changes. As global warming has its effects on local ecologies, we might expect that these rains come later and rains end earlier, which narrows the bell, and you just get a much higher but narrower season. You could also expect that there will be these dry mid-January spells, and we have a rainy fall and a rainy spring. We're experiencing one of these January conditions right now, where mushrooms are sort of declining in their diversity and abundance when you might normally expect them to be peaking, at least in decades past. So we'll see how these patterns change through time, but we're only going to see that if we manage to figure out a way to sustain these kinds of long-term monitoring efforts, not only here, but throughout the state and elsewhere. I mean, I would like for it to be done anywhere where there's a community of people interested in doing it. And now I want to talk about this sort of corny idea, an elemental view of these lessons. Water, earth, wind, and fire. Water is sort of the obvious elemental influence on mushroom fruiting in abundance. And that's in the form of rainfall. When it rains, mushrooms happen. It's the single greatest determinant of diversity and abundance on any given survey. So if it has rained, there's going to be mushrooms. And if it hasn't rained, there's going to be way less. So rainfall, at least in some sense, is the greatest determinant of diversity and abundance, followed by the habitat. And that's sort of generally wrapped up under this idea of earth and soil. The trees that are present, the soil type, the composition of the duff is what structures the diversity of mushrooms. And then they wait for rain. So to say that either of these is more important than the other is not really accurate. But between these two, these are sort of the most important drivers of what you're going to see on any given walk in the woods. So the soil can be all right and perfect, but if it hasn't rained, you're not going to see mushrooms. And if it's rained, but you're out in a concrete jungle in the urban setting, you're not going to see mushrooms either. So you need both appropriate habitat characteristics and recent rainfall to see mushrooms above ground. But these are sort of obvious. Everyone sort of understands what I mean when I talk about the influence of rain and habitat on the mushrooms that you see. But it's the latter two, wind and fire, that I actually learned a lot more about by doing this long-term, intensive, repeated monitoring. Wind turned out to be a really important driver of large shifts, big swings in abundance of diversity on very short timescales. So from one day to the next, there would be a big change in what I was detecting on a given transect based on whether or not there'd been a windstorm overnight, or one of those big offshore wind events where you had sustained dry, low humidity, 30-mile-an-hour winds. And this is likely a major selective force on the timing of fruiting, so when mushrooms produce their fruit bodies, and then what those fruit bodies are shaped like. So this is, when I mean a major selective force, I mean over evolutionary time. We often think of bugs, that sort of mushroom grazing predation, as potentially a driving force in the evolution of mushrooms. But I think humidity issues, how a mushroom keeps its fruit bodies hydrated long enough to produce ripe spores, is actually a major driving force. And it's sort of the difference between a little tiny mycena that's smaller than your pinky and a big bolete that might weigh 2 or 3 pounds. They've obviously taken very different evolutionary trajectories in how they want to invest their tissue in producing spores. So when I say that wind causes big swings in abundance and diversity, I mean in relation to mushrooms like this Mycena galopus that fruits all over the redwood litter when it's at the wettest point of the season. When the air humidity is high, when the duff is really wet, you can see thousands and thousands of these on any given day. But if you have a strong wind event overnight, these can all be wiped out within 48 hours. They'll all shrivel up and stop producing spores. So that kind of major fluctuation has much more of an impact on these small fruit body mushrooms that produce large quantities of fruit bodies than it does on a more robust fruit body that has a lower surface area to be dehydrated. So bolete is going to get through a wind event much easier than a mycena. And that's probably one of the reasons they produce so many fruit bodies, these mycenas do. Another example, so here's a close-up view of those mycena. Very slender, very fragile, relatively few spores produced on any individual fruit body compared to a big bolete mushroom or a big agaric like an amanita. But there's so many more of them that they make up for it that way. That said, they're more susceptible to wind. So the triggers that they use to fruit are probably going to be different. They're going to key in on extreme high humidity conditions. Here's another example of a mushroom that fruits in vast quantities and has relatively low surface area per fruit body, but as a whole, the individual might be producing a really large surface area. So this is Macrotifila juntia, or what we call that anyways. And as you can see, each fruit body is really just as simple as it gets. It's an upright stick covered in spore-producing cells. But what this means for this mushroom, which might weigh, hundredths of a gram, I don't even know how I would weigh this. I don't think my scale is accurate enough. it dries out instantly if there's any sort of wind. It's all surface area. There is no volume to this fruit body. But like I said, it gets around that by producing large numbers of individual fruit bodies. So it's this sort of clade of mushroom. It's not a clade. It's a ecological guild. It's a morphological suite. These mushrooms that are abundant but really slender and small. They have high surface area to volume ratios. They dry out really quickly and are super susceptible to the impacts of wind. But now I want to talk about fire. This is sort of the most interesting thing that happened to my transects in the time that I've been studying them. The CZU fire came through Santa Cruz in last summer, summer of 2020, after a lightning storm that was sort of totally unusual for our area at that time of year. We rarely get lightning in Santa Cruz ever, but to get it in the middle of the summer when the vegetation was sort of at its driest point was pretty catastrophic. It ended up burning over 86,000 acres at very high intensity and burned almost all of Ben Lomond Mountain, which is where most of my transects are. Most of the habitat type affected was mixed evergreen forest, including the old growth redwoods of Big Basin. So this was this extremely special, rare, southern disjunct patch of large old growth redwoods, not just redwoods, but some of the larger trees in this part of the state. And here's some satellite view of the CZU fire, and you can see just what a massive area it covered. I think 25% of the county's land area was burned in this fire, and you can see that scar affecting the entire north and west slopes of Ben Lomond Mountain, all the way down to the ocean. In fact, it burned down to beach sand, all the way from above 2,000 feet all the way down slope to beach sand. And for this is what the forest looked like after the fire in some places. So these are mostly Douglas fir and tan oak here. And you can see the soil is totally turned to ash. It did not leave any litter present in a lot of areas, and the trees are burned all the way up to their crown. So it was a crown fire. It didn't just stay low. It wasn't an understory burn. This was an intense inferno, tens of feet, 50 feet, in some cases, 100, 200 feet high. Here's a picture from the core of the old growth redwoods after the fire. I think this was taken in early November, before we'd really gotten any rain. You can see there's just a few stump sprouts on the tan oak tree in the middle of the photo there. And one of the things that redwoods do is they do this epicormic sprouting. They're actually quite resistant to fire, although they're not going to look the same for quite a while. They're still alive, and they'll produce these new photosynthetic twigs relatively rapidly after being burned. So a lot of the forest up there looks like this right now. A lot of stump spreading. So at the left of the photo there, that's redwood stump sprouting, resprouting after the fire. And in the right side of the photo, that's tan oak, which I didn't realize was such a vigorous regrower after fire. The stumps of tan oak trees can really resprout very vigorously. Unfortunately, tan oaks were already sort of threatened by sudden oak death syndrome, so we'll see how well they do after this sort of stressor event. They're not going to be tall trees again for quite a while. Their stump sprouts are quite short. So we'll see. I'm very curious the progression of this forest over the next 30 years or so, 50 years. But this is just what happened during the CZU fire. What happened to the mushrooms is the question. 4 of my transects burned last summer, which included some of the most diverse transects. So these were the transects that were affected by fire in 2020. And so far, all of them have had massive declines in diversity. At this time of year, I might expect to see 60 or 70 species on a transect, on any of these transects. And right now I'm detecting about 10, 15, 20 species. So really dramatic declines in the number of species. But that doesn't really reflect the whole story. You really expect that. That's what you'd expect after a fire, is that mushroom diversity goes down in the short term. But I want to know what the long-term picture is. And it also obscures the fact that even though the diversity and the abundance have gone way down, the abundances of some species have gone way up, and the diversity has shifted. So some species that were never detected before fire are already being detected on these transects immediately after the fire. So I want to give you some examples of what that looks like now. You've all probably seen this mushroom, Coloriza umbonata, the redwood rooter. It's present on my transects, So between the two survey seasons of 2018-19 and 1920, I found on this one particular transect, I found this species 6 times, and counts of that species range from 1 to 7 fruit bodies. So not a very abundant mushroom on this transect, about 1.8 fruit bodies or 2 mushrooms every time I went. But immediately after the fire in 2020, when I started my 2021 survey season, the first time that I found it on that transect, there were 125 fruit bodies. The second time I found it on that transect, there were 493 fruit bodies. So this is like a 70-fold increase from the prior high count. So this mushroom clearly really likes fire, which was not something that was known about the ecology of this species before. Here's just an example of how profusely it's fruiting right now. This is an area that's probably 1 meter by 1 meter square. And this fruiting, when I visited, had 155 fruit bodies. One individual, one little patch had 155 caps in it. Then there's the fire-following species like Anthracobia. The landscape is covered in these little cup fungi that specifically respond in response to fire, or fruit in response to fire. This polypore loves all of the burnt conifer trees that have fallen in the forest. This is Lentoporia carbonica, and it's one of the dominant species in the burned old growth redwood forest right now. And then there's the total mysteries. This is one of the most exciting things that has happened to me ever in the history of my mushroom hunting. On one of my recent visits to this burned area, I found this, and I have no idea what it is. It doesn't fit into any known genus or family that I'm aware of, and it fruits in these big clusters, and it was present in astonishing numbers, tens of thousands of fruit bodies, literally. I had to stop counting after 10,000. And I estimate those by chunks of 100, but I was immediately overwhelmed by how many there were. So this mushroom appears to be an unknown fire-adapted species that I had never seen anywhere before. It doesn't appear to look like any other genus that I know of. We're still trying to figure out what it is. I'm sending it out for DNA sequencing probably Monday morning. It appears to be a new species, a fire-adapted tannock-associated species. And just to give you a sense of how abundant it is, this is a picture of an area about 3 meters by 3 square meters. And I counted individually 294 caps. And this was just a 3 meter square area. This extended over the landscape for kilometers. So this mushroom is fruiting in gargantuan, colossal, titanic quantities in Santa Cruz County right now. Now we just have to figure out what it is. So all of this work was supported by a grant from the Save the Redwoods League. And I just want to thank them for taking a risk. Normally they fund sort of more obviously directly redwood focused research or botanical research. And they were excited to take this project on and fund it for 3 years. And even though it has been really intense and it's really taken a lot of effort, I'm really glad to be doing it. I've learned so much from this sort of repeated intensive visitation schedule. So I encourage you all to think about setting up long-term monitoring projects with your club or yourself. It doesn't have to be a kilometer long, and you don't have to do it as frequently as I do, but you'll learn a lot from doing repeat visits to the same area instead of just chasing the rains around. All right, so with that, I'm gonna quit sharing my screen and take questions from y'all.
All right, Christian, I've got some questions for you here. Marissa Gomez asks, do you have any sense of how the macrofungi diversity compares to, say, plant diversity?
On those transects? If the question is on those transects in particular, the plant diversity is actually quite low on most of my transects because they're in redwood forest and there tends to be almost no understory diversity. It's just a few, violets and a few other forbs. So on the transects that I'm doing, the mushrooms far outnumber the vascular plants. I don't know if that was exactly the question.
Sounds about right. Got a comment here from Jennifer Reisinga. She says, you're correct that the academic methods are difficult to replicate. I suspect that is intentional, but the crowdsourcing possible with citizen community science is going to change the utility of overly controlled parameters.
I can't agree more.
Amy Patton says, Christian, I like your shirt.
It's blooming. There's bees coming to me.
And Kenichi Ueda asks, is the impact of wind on fruit body abundance something you quantified? correlation to be determined. Seems like you'd need to be monitoring right next to a weather station to get that data unless you set up your own.
That's a really good point. So that was something I didn't anticipate about this project. And if I were to redo it, I would much more closely track wind. So I would like to quantify how much wind there was on a transect, but the sort of granularity and time that you'd need to like get the best form of that data isn't something I could really accommodate. So it's sort of one of the anecdotal results from this study that I would hope would structure future studies, that someone would really take a close look specifically at that variable.
Lizette Ariano asks, can you hypothesize a bit about the role of fungi post-fire?
I mean, so one of the obvious things is that they attract birds and insects, or not in that order, insects and then birds. So these mass fruitings where there's tens of thousands of fruit bodies all at once, they're also swarming with fungus gnats and other invertebrates that are coming to chow down on them, which means that birds are chasing them there. And if those birds are defecating any seeds or something, that's sort of an obvious pathway for recolonization of plants, is just sort of bringing in those dispersal vectors. But then there's the fact that mycorrhizal fungi, if they are still present, are going to be a crucial spore bank in the aftermath of the fire as new seedlings germinate there. They want to meet their mycorrhizal partners as soon as possible because they're sort of critical to scavenging nutrients and water. So they definitely play a big role in the recolonization of new plants. But then there's also the matter of all the dead wood that's out there now. So polypores like Lentoporia, and I think I'm starting to develop a hypothesis about Chlorrhiza as well, are kinds of fungi that respond to the fact that there's now just oodles of dead wood of all sorts, both hardwoods and conifers.
All right. Got a lot more questions coming in. You're a popular presenter, Christian. Eva Tadelman asks, what was the most interesting mushroom you found?
I think it has to be that last one that I mentioned that I just have no idea what it is. It was like, I got to my transect, I got out of the car, and that was the first mushroom I saw that day. And I had no idea. It was like seeing a zebra. It was like, what is this thing? I had no immediate reaction. And then I took it home and I did some microscope work and it still didn't help. So it was not only like seeing a zebra in the woods, but there was also 10,000 of them. It was like being surrounded by a UFO. Especially in the context of this fire ecology, it was a very intense and odd experience that I really enjoyed.
Well, it must have been a pretty wild specimen if you didn't recognize it. We'll go out on a limb and say that. Maya S. asks, you mentioned a club. Which ones are there?
Okay, there's a whole bunch of clubs. If you go to the Bay Area Mycological Society, there's like a Clubs of California Resources page. Their website lists them. But there's the Bay Area Mycological Society, which is in the East Bay. There's Fungus Federation of Santa Cruz in Santa Cruz. There's MSSF, Mycological Society of San Francisco. There's a Peninsula Club. Of course, none of them are meeting in person right now. So, get your shots and see you there next year.
Christian, how's your stamina for answering questions? We've got, they're still just flooding in.
As many as you want to read.
Great. Anna T, who I suspect is Anna Towers, our photographer, who's going to be sharing her some photos with us in just a moment, says, hello, how does fire assist in mushroom growth?
I mean, for some of them, it clearly provides tons of resources. So like all the dead wood specialists, there's now tons of dead wood. Some of it is chemically modified in a way that might make it more available to species that had a hard time colonizing the wood while it was alive. Obviously, there's going to be a big nutrient shift in what's available in the soil after fire. And I don't exactly know. There's very little data about what fire in coastal redwood systems does to fungi. We actually have quite a bit of data for Sierra Nevada systems because fire is more routine there. And the people at UC Berkeley studied it for a while. They also studied the effects of fire on fungi at Point Reyes. But as far as data for redwood tan oak mixed evergreen forests after fire, I couldn't really find much of fungal literature on that. There was a big fire, I think it was called the Creek Day Fire. I forget what it was called, up in Humboldt in the early 2000s. And I asked a few people about whether or not there had been any sort of targeted fungus data collection after that fire. And the answer was basically no. So I don't know. I don't know what's going to happen.
All right. Autumn S asks, can you speak about temperature and fruiting?
Not much. I know that for people who track things like that was, okay, Maria said it, the canoe fire. That was the name of it. I forgot what it was called. Thank you, Maria. Like people who hunt morels talk a lot about sort of the ascension of springtime soil temperatures before the production of those big flushes of larger ascomycetes. But I've never really paid too much attention to it because I'm not hunting for those edibles in such a sort of focused way. Usually I don't really care what the soil temperature is other than it's not freezing. But it would be really curious to keep closer track of that, especially in a burned area where it's probably much less stable, much more variable than in sort of an intact soil ecology.
All right. Lewis W asks, are you taking any soil samples on your transects?
I'm not. I tried to make this easy on myself and failed. So I'm trying not to add more work to it. I want this data to be a sort of a motivation for people to do that sort of more intense, highly quantified, single variable investigation. But yeah, I wouldn't have anywhere to put all that soil.
Fair enough. This is a good one. This is kind of a meta taxonomic question. Maya S asks, do you have a way to know which are known mushrooms and which are not?
That's sort of the work of daily life for me, is like, what is this thing? I don't know. What is that thing? I don't know. No, the answer is you just have to try and learn as many as you can. And then when you run up against one you don't, it doesn't mean that it isn't known. It just means you don't know it. And then if you work on it for really hard for a while and you still don't come up with an answer, then you make an argument to other people why it's not a known species. And sometimes you're right and sometimes you're wrong, but hopefully you do your due diligence, do your homework, and don't publish a bunch of spurious new species, but it's happened. Not to me, of course.
Of course not. Of course not. Matthew K wants to know, do we have any ideas about how to start classifying new species without standardized academic venues as the multiple sequencing and ID information is becoming more widely available?
I'm not exactly can you read that one more time?
Do we have any ideas about how to start classifying new species without standardized academic venues? I'll just end it at that.
It's always been possible for non-academics to publish new species, and it's actually becoming more common. They pretty much go about it the same way that academics do, and in some ways I think they do it better on average, at least in the world of mycology. Yeah, there's no guarantee that just being an academic means that you're a good taxonomist, and vice versa. So it happens. It's actually, if you have access to the kinds of tools that are sort of desired for new species descriptions like DNA sequencing, which is actually becoming more and more accessible to amateurs these days. If you can do that, you can make a description of a new species without any sort of institutional affiliation.
Well, there you go. Even quote unquote amateurs can describe species. All right, Jessica N wants to know, is there a name for the two types of mushroom strategies you mentioned, the many/big surface area versus the few/robust? Isn't that kind of like R and K strategists with mammals?
Yes, but there's some important differences, because when we're talking about R and K with mammals, we're talking about the offspring that has the potential to grow into an adult. But what we're talking about with mushrooms is the fruit body that itself produces propagules. So there's no adult involved yet. There's no investment in an individual. It's an investment in the propagules. So mushrooms are sort of fundamentally low investment in their offspring because they produce these tiny little spores, most of which die. And that's true of a bolete or a mycena. So we're talking about a slightly different sort of further back concept, but it's very analogous in some ways. But I don't know if there's specialized terminology to talk about those two strategies. Someone probably has, and I just don't know it.
Well, maybe you could coin it. Athena J wants to know, do you have a theory for how the long-lived Gymnopolis fruit body benefits from the long fruiting time?
It just increases its sporulation period. So it's a way of increasing your surface area through time rather than through space. So some mushrooms take I'm going to fruit huge numbers just for a short time, and so they have a high surface area, but it's temporarily limited. Other mushrooms do the smaller surface area, longer persistence, so the surface area has this weird 4-dimensional persistence. So I would imagine it just lets it make more spores.
Well, there you go. Haley D wants to know, in the places where the CZU fire burned most intensely, topsoil totally charred, were all the below ground fungi destroyed?
Probably not all of them, no. There's actually an amazing insulating effect of even a few centimeters of soil. And I've found mycorrhizal fungi already fruiting in some of none of the most burned places yet, but in some places quite closely surrounded where clearly the soil got hot. There's still mycelia there. So just because the fire came through, even intensely, doesn't mean that everything died. In some places it did, but not everything.
Interesting. We're almost few. We're down to the single-digit questions left over now. So thanks, Christian.
Sure.
What a guy, huh?
These are good questions.
Brett M wants to know, did you see a shift in species on the unburned surrounding edge around the fire as well? Ooh. And was it different from the change you saw inside the fire area?
That's a really good question that I actually haven't analyzed the data for yet because I'm still in the process of collecting it. But anecdotally, it feels like yes for some species. So I had this sort of, I don't know, I might be deluding myself, but Coloriza, that redwood rooter mushroom, clearly had a huge boost in the burned areas. I haven't tallied up the numbers yet, but it feels to me like it's also more abundant in unburned areas this year as well. And I wonder if there's some response to the fact that smoke particulate precipitated out and it uses smoke as a trigger. Or I know this is the case for some plants, but that sounds like a little bit far out. I don't know if that has any sort of realism behind it or if I'm just deluding myself, but that's what the data analysis is for. We'll find out later.
Okay, great. Let's see here. Haley D wants to know if you have any plans to continue, or if there are any plans to continue monitoring fungi and post-fire ecology in the CZU area.
I didn't know this was going to happen to my transects. It wasn't set up as a fire monitoring study. Obviously, my data got way more interesting because of it, because I got sort of the last 2 years of baseline data that were possible to get out of Big Basin before 95% of the park burned, including pretty much all of the mixed evergreen. So I desperately want that data to continue, but I don't necessarily know that I'm going to do it.
Well, fair enough. A follow-up question to that, Anisha K wants to know, is there somewhere we can find a guide to do long-term monitoring in our area?
This protocol is super simple, and I have it written in a little PDF that I think what I will do is make a blog post about as a follow-up to this. And I don't know if I sent it to Trent, would there be a place you could put it for your attendees to follow up on later?
Yeah, we could probably link it to the video that's going to be hosted on YouTube, and we can post it to the Facebook account.
Boom, there we go. Okay, so that's what'll happen. I'll post the materials that I have about the protocol for this, which I made up and may not be very good, but they're what I made up, and explain sort of my rationale for the different steps, and then Trent can link to it in the follow-up videos that are posted from this event.
Yeah, sounds great. Inspiring people to do more citizen science.
That's the idea. That's the hope.
A couple more project-related questions. Carolyn B wants to know, what direction do you anticipate this project going in the future? Kind of a similar question to before.
I just want there to be some sort of persistence. I mean, if it ends up being the case that it's really hard to find someone to do this, because it does take time and a little bit of sort of a niche expertise about being able to identify a lot of mushrooms. If it's hard to find someone to do it in the near term, what we can do is we can reduce the number of species, the number of focal species, so that you only have to be able to identify this sort of core set. You don't have to worry about counting every mushroom. We can reduce the intensity, so fewer transects. I don't think we should reduce the frequency, because the frequency actually had a lot of really, really high value. But what we could also do is we could have, an off period where we sort of don't collect data for a while, and then we come back 5 or 10 years from now and recollect data and compare at longer intervals. I would prefer it to be sort of contiguous, but resources are limited, not least of which is people's time.
Right. This Kenichi guy keeps asking questions here. He's got another one. Are the abundance and diversity patterns you observed mirrored in the overall iNat, that's short for iNaturalist data for Santa Cruz County, if you subtract your own observations?
That is literally the thing that I'm going to be focused on comparing once all of this is over, is I'm going to take all of the iNat observations from my county, subtract my own, and then see if the scattershot, haphazardly collected citizen science data with no protocol is a pretty good substitute for my ridiculously intensive data. Because if it is, then I want to quit doing what I'm doing, because we're doing good enough by just being a community of people sampling at random. Obviously, there's going to be patterns that I was able to capture that the general community wasn't, because it's just more intensive. But if we get most of the value from unstructured data collection, then that's probably the way to go in terms of using resources effectively. But it's also just going to be interesting to see if that works. I mean, there's things that the general community captures that I don't, because I'm restricted to certain kinds of habitats.
Right. Restricted to cool, dank, wet habitats like your fungal friends. A few kind of general ecology questions here. Well, one more related to fire. Daniel wants to know, are these mushrooms fruiting so prolifically this year because of the fires? Do they sense a danger to their current habitat and want to try to migrate away to find a more fertile area?
That's definitely the hypothesis for things like morels, and probably those fire-adapted ones that I mentioned, like Coloriza and that weird undescribed thing that I don't know what it is yet. It certainly seems like something that they're doing is that whatever their ecology was, it ran into a limiting sort of end. Maybe their host died, or maybe there's this sort of soil change where they want to reproduce really quickly, put propagules out to find new safe areas, because their mycelium may die out in that area after the fire. That's certainly the hypothesis for morels in many places. I don't know what the current research is on it, whether or not there's much persistence after the fire of those fire respondent species. I don't know.
Okay. Morgan C. wants to know, do you know much about underground competition amongst mycelia between species or even organisms?
The answer is no. I wish I did, but I don't. It's really hard to study because it's hard to see interactions between mycelia in situ without killing them. So it's not an easy thing to study. A lot of the case studies that we have are actually in wood, where you're able to sterilize a piece of wood and artificially inoculate it with two known taxa and watch them grow together and watch those interactions over time. You can make cuts through the wood and sort of like kill them at different points to see what happens. That said, I don't even know that much about those. I downloaded a paper yesterday about the control of bacterial communities in rotting wood as shaped by fungal variables, but I haven't read it yet.
All right, one more from Morgan C. What's the process to officially define a new species?
There's not one process. There's lots of different avenues you can take, but basically you come up with a name, you make an argument for why it's new, you assign a type specimen. So that's really crucial, is that there's an actual physical representation of the new name that you're proposing. And then you put it out in the world in some sort of sanctioned way. It has to come with, it has to be in a particular kind of publication that's recognized by the community. Now, that sounds like it might be some sort of hurdle, but actually they kind of just let any Yahoo publish these days, which is not great. There's varying levels of peer review. You can pay a whole bunch of money and still have really crappy peer review, or you can write a really great paper that never gets peer reviewed and still publish that way. It's kind of a chaotic and definitely not ideal landscape, but ain't that the human story?
Indeed. I think I'm going to end the questions with this one. Kelly K asks, my 5-year-old wants to know, what cool name would you give to that mushroom if it's a new discovery? That's
awesome. Your 5-year-old knows what I was doing at 2 a.m, lying awake with insomnia. I was like, this mushroom's too cool to not have a cool name. What would I call it? I was thinking Cinereosabe, which means ash caps in Latin. And then I was thinking for the species name exuberans, because it's fruiting in such large numbers. So the Latin translation would be something like exuberant ash cap.
I like that. The exuberant ash cap mushroom. Christian, I'm gonna ask you one more question for myself, and then we're gonna take a short break here. But I was wondering if you could speak on California's rapidly changing rainfall patterns, specifically our compressed rainfall cycle, and the sort of overall effects on fungi, particularly, Dicaria or large fruit body bearing fungi.
Yeah, so it certainly seems, I mean, I'm not an expert on, not even close to an expert on the rainfall regime changes. Daniel Swain's blog is where you want to go if you want to read about this and see the links out to other research about it. But my general anecdotal impressions line up with what he has been talking about, which is that fall rains are coming later. Spring rains are sort of dropping off quicker. So we have this highly compressed core rain period. That's sort of the only remaining reliable rainy season. And it certainly feels that way with mushrooms. A lot of years we're getting what we normally thought of as fall associated species overlapping, sort of like stepping on the toes of winter and spring species all at once. This year is a great example of it. In the past week, I already saw some fall associated boletes fruiting at the same time as what I think of as late season waxy caps. At the same time that fetid adder's tongue is blooming, which, fetid adder's tongue is just a little bit early this year. That's about normal, but we almost never see it at the same time as those fall boletes. So that just felt sort of phenologically jumbled to me, like everything is happening at once.
Right. I asked that because, I don't have any hard data to support this, but anecdotally, what I've noticed here in the East Bay is that our ectomycorrhizal fungi, the season is just kind of becoming compressed and happens much, much later.
Yeah,
common species like Russula rhodocephala, which normally we would find in wild abundance after our first fall rains, still aren't up in Tilden nature area right now. So
yeah, I mean, that's exactly right. Like, to try, if you were to rewind the tape back to the year 2000, I think the data show that it would be hard to find Cocora, that big, beautiful Amanita calyptoderma. It would be hard to find it in January a couple decades ago. And nowadays, it seems like you can find Cocora in January every year or every other year. They're fruiting in Santa Cruz right now, which is really late for them. Normally, we expect to see them in November.
Right. Interesting. Well, wild times if you enjoy changing weather regimes. Christian,
thank you. Don't.
Well, if you don't,
they're still wild.
They're still wild, and you're kind of stuck with it. Yeah. Thanks so much, Christian. That was a fantastic
thanks for having me.
Yeah, and folks, I've asked Christian to stick around to help me review some of my extensive collection of mushroom field guides. I thought, who better to help check out these different field guides than a field guide author. So we're going to do that in about 13-ish minutes. We're going to take a little break, let everyone, go get your drinks of water or go to the restroom or whatever you need to do. Stop looking at your computer for a few minutes. If you want to keep looking at your computer, however, I've dropped one link into the chat box, and that's the link to Daniel Swain's weather blog, weatherwest.com. It's fantastic. And I'm going to go ahead and paste in, again, that iNaturalist link, which will take you over to our iNaturalist MycoBlitz event page, where you can peruse all of the fungi that are fruiting right now in the Greater Bay Area, or at least all of the fungi that have been observed by humans. So check out those two links and take a little break. I'm going to queue up a slideshow here while we're on our break. It's going to feature the mushroom photography of Anna Towers. You can find her on Instagram at The Friendly Fungus. Anna's a local photographer who's been at our fungus fair in person since we began hosting it. She's the one that's usually set up at a table wearing a really great Amanita muscaria hat. But again, since we're virtual this year, please enjoy this slideshow of her photography. And we will be back at 11:15 to review mushroom guidebooks. All right, welcome back everyone to the 2021 Tilden Fungus Fair. I'm your host, Trent Pearce, broadcasting from Tilden Nature Area in Berkeley, California, part of the East Bay Regional Park District. We're going to get right in now to a naturalist's guide to mushroom identification books. Joining me is mycologist and author Christian Schwarz. Thanks for sticking around, Christian. That was a lot of talking. Appreciate you still being here.
I can't here.
Let's get right into it, folks. I'm going to switch here to my handy dandy field guide camera. And, you know, Christian, I just wanted to go ahead and start with this one because, well, this is the one. This is my favorite field guide that's really ever been written. Not to put too fine a point on it, but it's called Mushrooms of the Redwood Coast, and you are the co-author along with Noah Siegel. Did I say his last name right?
Yep.
Great. I guess I want to ask you some questions about this. So what's it like writing a mushroom guidebook? Specifically, how did you decide to organize it? I mean, when there's so many other guidebooks out there, what was your sort of process on laying this one out?
Well, our first motivation was that we wanted to just sort of update what was available for California mushroom identification guides. There wasn't one that had full color photos for every species covered. And there was also just a lot of research that had been done in the intervening years since Mushrooms Demystified, which was the last major field guide for California. And so we just wanted to sort of make a field guide that was modern in both of those senses. More understanding of some groups of species that weren't well known before, and better color photos, more color photos. So that was the motivation. And we were really writing it for ourselves because we wanted sort of something to consult for, because I can't keep all this in my head. So I want to do the research, make it concrete in a book, and then I get to use the book too. So I was writing it for me as much as I was writing it for anyone. And then as far as organization, we wanted to organize it visually so that the most similar looking species were right next to each other, regardless of whether or not their names were similar, regardless of whether or not they were closely related, because we found that that's what people confuse is mushrooms that look alike. You know, it's not, that's sort of the most important variable is the visual similarity. So we organized it by visual similarity, more or less. There's a few other variables that we use to structure the book, but that was the major one.
Great. Well, it's a fantastic book. I mean, I think folks who, you know, first pick it up are probably immediately struck by the photographs. They are unbelievably good. And I particularly appreciate that, you know, each sort of species has its own photograph that isn't just a little snapshot, you know, of a mushroom in situ. As much as we all like to leave things in situ sometimes and photograph them as they were, you and Noah kind of laid out each of these photographs in a way that you have a selection of each representative, you know, species in various forms of growth, and you know, and posed sort of just beautifully in their habitat. I mean, it's just the best.
Yeah, that was taken almost directly from Bill Rudy's Mushrooms of West Virginia, I think was the name of that book. He had this really cool style of photography that Noah always really admired. And that was sort of one of the big field guides that he used when he was learning. And he wanted to sort of bring that west and replicate it here. Steve Trudell was also a big influence on the photography style. So we just sort of followed their lead. And then, of course, now it's Noah really developed his own style, you know, taking off and modifying both of those two that I just mentioned. And I learned a lot from him. I mean, I didn't know how to photograph mushrooms when we started doing this book.
Well, you certainly wouldn't know that by looking at it. Now, just ballpark, how many of the species in here do you think you personally photographed?
You know, I've never counted. It's Noah has the majority, but I don't know if it's like, I don't know what the breakdown is. I can remember them all, though. So if you point to one, I'll tell you whether I think the middle one is mine there.
Okay, let's see here. Let's just flip to a random page with a big color photograph. There we go. Hygrosity virescens.
That's mine.
Oh, nice. Of course, with the
I've seen Mark's 2014.
Yeah. So for our viewers out there, I'll have to say I've had the pleasure of going out into the field with Christian many times, and I've watched him take pictures of mushrooms. And
you mean you've endured it?
What's that?
You mean you've endured it?
I have endured it. Well, you know, I think anyone who is a natural history every photographer understands that it's a process, and that if you go out with your friends who aren't photographers, they're going to get really bored pretty quickly. Luckily, I'm a photographer myself, so I've always appreciated watching Christian stage these sort of beautiful photographs. And also knowing that, you know, they may contribute to actual science in the future, unlike all of my pictures, which just end up on my Instagram to garnish a few likes. I feel compelled to point this out to our viewers too. I have this fantastic bookmark in here, which is probably the best business card I've ever seen in my life. Of course, that's your business card, Christian. I use that to mark whatever thing I'm looking at in the book.
Oh yeah, there's actually a question in the chat right now that's a good one to address.
Oh, great.
About the identification sort of structure of the book. So Mushrooms Demystified, for those of you who have used it, is probably the best mushroom book for beginners, total beginners to the world of mushroom hunting. So I really still highly recommend Mushrooms Demystified. It's, I think, one of the best introductory field guides to any group of organisms out there. But the way that identification is structured in Mushrooms Demystified is as a dichotomous key, where you take pairs of descriptions and you choose which one of those descriptions sounds more like your mushroom, and you follow this branching tree of decisions until you get to a name. And we decided not to do that in Mushrooms of the Redwood Coast. Since we arranged it pictorially, we started with, on page 17, this pictorial guide to mushrooms. So you just choose which of these sections your mushroom looks most like, and which of those paragraphs it matches most closely, and that takes you to a group. So someone just asked who's the author for Mushrooms Demystified, and the answer is David Aurora.
Yeah. And we'll get to that. Don't worry. I would be very remiss to not cover Mushrooms Demystified in a Naturalist's Guide to Mushroom Identification books.
But anyways, once you've used this pictorial key to choose your section, it takes you to a beige page. So those are these chapter opener pages. And then you just sort of read the background for that group. So any example will do. You go to a beige page and read about that particular group. And as soon as you've done that, then it's just a matter of flipping through and picture matching. It's just a matter of visually comparing mushrooms.
Right. So, for example, here in our pictorial key in the beginning, we've got russulas and spores white to cream yellowish ochre, page 227. And if we go to page 227, here's the aforementioned beige guide.
The first step is you read that, and then you just start flipping and look for something that looks like yours. So let's say we just stop on one, say the Russula cremoricolor there. If you've gotten to something that you think looks good for your mushroom, you don't stop, flip one or two more pages to make sure there's not an even better match one or two pages further. Oh, so let's imagine we flipped one more page and we're like, oh yeah, Russula rhodocephala, that's what we're talking about. That looks perfect.
I've updated the taxonomy also with a post-it note.
Oh my God, I'm so proud. So now what you do is skip to the comments section. So people often think that reading the technical description is the first thing, but actually the comment section is the first thing you want to read in our book, because that will tell you what you're likely to have misidentified it for and how to tell those things apart. So it's a little cheat sheet. And then you skip to the ecology section, which tells you what trees you should be around and which part of the coast you might find it on. And if those don't match, then maybe you want to be a little more careful about your ID and try a few others. And only then do you really go to the technical section if you need to. But I really recommend sticking to the comments and the ecology section first and foremost. And then I always tell people the last step in using our book is not in the book. It's going and talking to a real person. And either posting it on iNaturalist, talking to people on Facebook, giving your friend a call who knows mushrooms, and getting that validation step. Because the last step in the learning process is the thumbs up or thumbs down from someone who knows what they're talking about, and feedback on what you did right or wrong. So the last step of the identification process is not in the book. It's actually talking to a real person.
Great. Well said. You know, I think for me personally, that's sort of one of the reasons I enjoy this book the most, is because it is the type of book that you can flip through and look at photos. And that's how you laid it out. And that's sort of how my brain works. I mean, I certainly have used dichotomous keys in the past, but this just works for me. And I think it probably does a lot of our audience out there. You know, they appreciate being able to go to a section and flip through and be like, oh, it looks like Russula mendesinensis. And then, like you said, reading the comments and then the ecology. Yeah, so folks, this is the book. If you don't have it, you should buy it. Christian, how can folks out there who are watching our program today best purchase this book so that they give the least money to the man and the most to you, or whatever?
I'm just going to put this in the link, but www.redwoodcoastmushrooms.org. is the website where if you order it from there, I'll be the one mailing it. Normally it would be at your event. I would be doing hand-to-hand sales, which is my favorite way because I get to meet people. But in the meantime, we'll just do it this way.
Well, in these trying times, we'll do the best we can.
I've stopped trying.
Yeah. Unprecedented. All right, so Mushrooms of the Redwood Coast. That's number one in my recommendation for mushroom guidebooks of California. But I guess number 1.5 would have to be this one. There it is, the wee little timeless, all the rain promises and more. So I learned California fungi with this book, or I initially learned them, and it was really helpful for me. Oops, don't look at Rick's phone number there. That's not supposed to be in there. This was a fantastic learning guide for me when I was a beginner back, a decade ago or something, because much like your book, it has a pictorial key here in the beginning that sort of guides you to these generic sections, right? And this, it's got little illustrations that asks you basic questions, gills, yes, no, breaks cleanly, has a vulva at the base, all these things that will guide you to sections. And then yes.
That guy's my neighbor.
Mine has been autographed by David Aurora. Sorry, what?
That guy's my neighbor.
Oh, nice. And then, the little pictorial guide in the beginning will take you to a section. So we flip to, the Amanita section here.
I should point out that this is the guide that I have the least experience using.
Really?
I got it because it had some of it's obviously a really fun book. There's tons of pictures of people and stuff. Mm-hmm. But the color pictures were sort of an update on what was available and demystified. So that's one of the main reasons I've used it. But I rarely use the part at the front, but I've heard a lot of my students say that they really appreciated that flow chart.
Yeah, well, like I said, it was very helpful for me when I was initially learning California fungi. And so we get back to a section here, and then sort of in the same way, we've got, as your book was laid out, we've got a generic sort of description of the group. In this case, it's a genus. And then we flip through and just sort of look at pictures until we find a match. Of course, however, unlike Mushrooms of the Redwood Coast or really any of these other field guides that I'm going to talk about, this is a very small and non-comprehensive guide, and it kind of only has these sort of very charismatic representatives of each group in it.
And it covers a large area, so there are mushrooms that you won't find in California represented.
Oh, that's good to know. Yeah, so sort of broad coverage, and as well, it hasn't been updated in, where's the published date on this? Last updated in 1990.
Yeah, but I have to say it's definitely one of the most fun and beautifully illustrated little guides, and its small size is actually something going for it. It's one of its main value. I think he advertised it as being able to fit in your pocket, in your back pocket while you're walking around the woods. That said, there's the necessary trade-off with it being sort of limited in coverage, but it is a really good edition.
Yeah. It's a fantastic book. Mine is dog-eared from being in my back pocket so many times while I was out hunting. And I'll say one more thing or a few more things about this book. It has fantastic sort of anecdotal stories in it, like my most memorable mushroom hunt. And here's a photo of, some chuck barrows with Craterellus cornucopioides, right? And here's this story that the author, David Aurora, has written about chuck barrows. So these stories most of them are hilarious. And they all, serve to illustrate kind of some sort of lesson about mushroom hunting or identification or whatnot. So great book. I still recommend this to people who are just completely beginning to learn fungi, particularly Western or California fungi, because it does have a slight bias towards those.
Yeah.
And just because it's very simple, and it will teach you to recognize these larger groups pretty easily. And then once you are familiar with this book, then you can, dive right into something like this, and you won't even need to use those guides. You'll just be like, oh, I have a russula. Now I need to look at Christian's book to figure out which russula I have. So that's All the Rain Promises and More by David Aurora, a hip pocket guide to Western mushrooms. And it is a timeless classic still to this day. Also, for our viewers out there, that's not David Aurora. That is a common misconception. That's not David Aurora on the cover.
I'm pretty sure he's a computer science. The person on the cover is a computer science professor at UC Santa Cruz.
Yeah, there are photos of David Aurora in here, but they're kind of cryptic. There's
the one with the toaster at the very front.
Yeah, the reflection of him in the toaster that I just had.
Or the honey mushrooms too.
Right. But anyway, that's not David Aurora. But the author is David Aurora. The book is All the Rain Promises and More. Worth having in your mushroom library. Okay, next up, of course, the quote-unquote Bible. Yeah, a lot of mushroom hunters call this the Bible. In my opinion, your book is sort of usurping that title. But this was the original comprehensive guide to really North American fungi, right? I mean, Aurora tried to cram as much as he could into this book. I've seen a lot of love. I actually took it out of its custom dust jacket for this presentation. So unlike the other books that we've looked at so far, this book is primarily text-based. It's got a lot of words, and each section, let me see if I can find one of these, has a very lengthy dichotomous key at the beginning of the section. So, okay, we've got an interloma, and then we're going to go to, here's the key to interloma, and then we've got these couplets, like you mentioned earlier, you know, cap red, brown, or not as above, and then you go to the sections. So if anyone out there is, you know, has experience keying out plants using something like the Jepson manual, this is kind of the same book. This is sort of the Jepson manual equivalent.
Although I will say one of the most amazing things about this book is how good David is at writing keys. So the Jepson manual, anyone who's used it is going to be like, oh, I don't want any part of that, because the Jepson manual is often difficult. There's an art to using a dichotomous key. You have to learn to use them. But David writes really good keys, especially for beginners. They're not technical. They're usually in like very plain language, and he really knows what features to key in on. So this was like one of the major innovations of this book that I think made it so revolutionary for people, is that they were able to learn in a structured way how to recognize different groups of mushrooms. So David's keys are amazing.
Yeah, I'd have to agree with you. Maybe that was an unfair comparison to compare it to the Jepson.
Oh, no. I mean, most people are familiar with dichotomous keys from other sources, so it was a good comparison. Yeah.
Right. So we've got our dichotomous key, and then we've got these very sort of lengthy, even verbose, you might say, species descriptions. And then occasionally we'll have a little black and white photo mixed in with the species descriptions. So if you're a person who enjoys buttery color plates, this book's a little bit lacking in that. It has a section in the middle. I think all my pages are still attached of, you know, what by now are practically sort of antique photographs. I mean, we know these were shot on an old 35 millimeter, maybe on slides. So in a way that makes these photos even more impressive that they were taken so long ago with such dated technology. But so there's a little sort of color plate section in the middle. It's not very thick. Yeah, I'd say it's, you know, less than a fifth of the book that has color illustrations or color photos of really only the most commonly found species, or the ones that David thought were charismatic enough to include in the section. This is still one of my favorite spreads, though, the hygrosabe and hygrophorus section, so pretty. So this is Mushrooms Demystified by David Aurora, kind of the original definitive guide to fungi, and still a very good book. Again, hasn't been updated in quite some time. I think it was last updated in '86. So the taxonomy in this book is going to be pretty out of date, but you can still go to a mushroom, you know, key it out and get to a species, and then you can just get online and sort of compare and see what we now understand as that species concept in North America or even California, right?
Yeah.
Christian?
Yeah.
Begley? Yeah. All right. So Mushrooms Demystified. If you get a new copy, the cover will probably be attached. Okay. This is like when I was in kindergarten and I got to do show and tell. This is how I feel right now. So here's California Mushrooms, the Comprehensive Identification Guide. This is by Dennis Desjardins, Michael Wood, and Fred Stevens. And this is a great book too, although in my opinion, this book is slightly mistitled. I think it should say the comprehensive, or it should be titled Sierra Nevada Mushrooms, the Comprehensive Identification Guide, because it really focuses on Sierra and fungi.
Yeah, one of the cool things about Desjardins and Stevenson Wood is that they were associated with the Sierra Nevada field campus of the SFSU sort of field school. So they had this amazing data set of years and years and years of many students going up there, really closely looking at fungi that live in the Sierra Nevada, right up around Yuba Pass, Bullard's Bar. And so a lot of those photos and a lot of the knowledge of the mushrooms of that area come from these, from their work. And also, for those of you who don't know, Mike Wood runs MycoWeb, which is this amazing online resource to fungi. And a lot of these same photos and descriptions draw from what's there. So it's years and years and years of work that Mike put into that website that then sort of were crystallized in this book.
Yeah. It's funny you mentioned that, Christian. I've actually taken a course with Dennis Desjardins up at the Sierra Field Campus, and awesome. I pulled this out of this book right before I got ready for the program today. This is the spring fungi list from 1982 to 2007.
Nice.
This is a handout.
That's a lot of decades.
Students. Yeah. Yeah. So it's a lot of data. So this book, meticulously researched, again, sort of biased towards the fungi of the Sierra Nevada, but in that way, it kind of makes an excellent sort of companion book to Mushrooms of the Redwood Coast, wouldn't you say?
Because we have absolutely no coverage of the Sierra Nevada.
Right.
I mean, there are a few mushrooms that we have in our book that occur there, but you don't try and identify mountain mushrooms with our book.
Right, right. And again, this is more of a textbook format. This is a big, heavy book with a hardcover, so it's probably not something you're going to take out into the field with you. This is really one that you go collecting in the Sierra, and you come back to your campsite or maybe your house, whatever, and you pull out this book. It does have a key at the beginning of each section again, so it's a dichotomous key, much like David Aurora's books. So we've got the different keys that will take you to the different pages, and then pretty much the same thing. You look at the photo, you read all of the description. The comments sections are quite lengthy in here. And
Debbie just pointed out that Fred Stevens is also involved in running MycoWeb, and that's true. And actually, his photos I think are really spectacular. I always enjoy Fred's photos, and he's also an agaricus expert, which makes him very near and dear to my interests. I learned a lot of agaricus from Fred.
Yeah, and MycoWeb is, I believe that was the resource online that I pretty much used for identifying California fungi with updated taxonomy until your book came out.
Oh, for sure. I've used that website so much. I mean,
it's a great companion to this book, or really to any guidebook. MycoWeb. We'll throw the link to that in the comments box shortly. Yeah. So this book, California Mushrooms: The Comprehensive Identification Guide. Big, sturdy textbook style book, coverage of Sierra Nevada fungi primarily, but a great book to have on any California mushroom hunter's shelf. So we've got just a few more minutes here. I want to delve just a little bit into the unusual. Here, Christian, you'll have to indulge me on this one. I was down at Pegasus Books in Berkeley, and I saw this on the shelf, and I thought, I have to have that. And it's because this was written over 100 years ago. This was first published in the year 1900 by Charles McIlvaine.
There was a lot fewer mushrooms back then. Most of them hadn't evolved yet.
Right. Yeah, it was an entirely different, mycoflora in North America.
It was called 1,000 American fungi because there weren't anymore. That was all of them.
This is literally all the mushrooms that were in North America. Yeah. So this, at the time of its publication, this was the comprehensive guide to all North American fungi. I mean, 1,000 descriptions, 182 illustrations. This book is sort of lovingly, illustrated by hand with these charming line drawings. For select species. But my favorite part about this is the fact that McIlvaine personally tasted something like 600 out of the 1,000 species described in his book. In fact, I found a quote that he was quoted as saying, "I take no man's word for the qualities of a toadstool," meaning that he didn't believe what people said about their edibility. He needed to taste them himself. And so his edibility descriptions for some of these are just kind of, bold, I would say. He taste tested and actually described the virtuous tasting of a lot of species that we today consider toxic. So yeah, this book, don't use it for an edibility guide, but
I like using it for entertainment.
Yes.
Like when you're at a post mushroom event dinner with your friends or a party and the wine is flowing, you can open to random pages and read his edibility accounts, and it's usually good for a few laughs.
Exactly.
And he's also an observant naturalist. Like his descriptions of mushrooms were actually pretty good. They really communicated the essence of the mushroom in many cases.
Right. Capturing the gestalt of the mushroom via words.
Not so much that one on that page. I can't tell what that is,
but something like that. But anyway, so this book is 1,000 American Fungi, originally published in 1900, written by Charles McIlvaine and Robert K. McAdam.
Which I feel like we never talk about McAdam. Who's I want to know more about him.
I don't have any information. I mean, one of our viewers out there should look up Robert K. McAdam and see how robust his internet personality is.
Deep dive on Bobby Mac.
Yeah, probably not a lot. Okay, folks, I'm going to touch on just a couple more here. I'll bet no one's ever even seen this book. Yeah, I just have this because I had one of my photos published in it. It's right here. It's not even a good photo. I think I took it with a cell phone. But nevertheless, this is Mushrooms of the Northwest. And so I actually wanted to feature this book for a couple of reasons. One is that we have pretty decent coverage of California fungi in terms of guidebooks. We have Mushrooms of the Redwood Coast. We have David's two books, which again cover more than just California, but are very California-centric. We have Desjardins and Wood and Stevens' book, which covers Sierra fungi, but all of those sort of end right somewhere around the Siskiyou Range in Northern California, Southern Oregon. So for any of our viewers out there that like to travel up to Oregon or Washington, there's a chance that you're going to encounter fungi that aren't described in any of the books that we have mentioned before. So I like to have in my library, a handful of other books specific to the Northwest.
Do you have Steve Trudell's book there at hand somewhere?
Yep.
So that's one that I really recommend for people in the Pacific Northwest. Steve Trudell is an amazing photographer. Joe Amorati is old school. He's been doing mushroom taxonomy and mycology academically for a long time. So a lot of expertise in that book, really good photos.
Yeah, this was actually the one I was going to feature, and I was afraid we were going to run out of time. So, yes, this is my go-to book for fungi that sort of show up on iNaturalist that are outside of our standard range of field guides that I have loosely committed to memory.
And I hear that there's a second edition of it in the works.
Oh, great! I love it when field guide authors update their books. Anyway. So this is a good book as well. Mushrooms of the Pacific Northwest by Steve Trudell and Joe Amorati. That's great coverage for basically where all of the guides that we've reviewed up to this point end. And since we've got just a couple more minutes, humor me here. I'm going to throw in something for, I know we had at least one viewer in the Eastern United States. So I'm just going to mention the Bassett books. These are really big, heavy books, but this is a series of books. So this is Northeastern North America. We've got Southeastern United States, written by Alan and Arlene Bassett, and then various other authors for each one. And these kind of have a fairly good coverage of fungi on the other side of the Rocky Mountains. Mm-hmm. And to my knowledge, they're probably still the definitive guide for Eastern fungi in North America.
I know that Tim Baroni co-authored one recently that I haven't had a chance to look at, I think from Timber Press. Obviously, I don't really use the Eastern guides that much, but it's one that if I ever, am allowed to travel again, when I go back east to go mushrooming, I'm going to pick up one of those. And Todd Elliott also, was involved. I don't know if he co-wrote the one with Baroni, if he wrote his own. Todd Elliott and Baroni. I think that's right. But they have a new one to Northeastern North America that I'm really looking forward to using.
Oh, great. All right. Well, maybe we can, maybe you can throw that into the comments section for our viewers out there. Yeah. Excellent. Well, that concludes A Naturalist's Guide to Mushroom Identification Books. Thanks for watching that, everyone. I am now going to turn it over to my co-host, Melissa, who is going to introduce our next guest presenter. Thanks again, Christian, for participating in the 2021 Tilden Fungus Fair and brightening everyone's pandemic-dimmed lives.
Sure thing. See ya.
All right.
All right, good afternoon, everyone. My name is Melissa Fowlks, and I am a naturalist at Big Break Visitor Center along the Delta in Oakley. And I'm so excited to be here today, and even more excited to introduce Morgan Evans to y'all. Morgan holds an associate's degree in culinary arts and loves to merge her two interests, the outdoors and food. A certified interpretive guide with the National Association for Interpretation. She enjoys backpacking, botanizing, and foraging for wild edibles. Morgan is a recreation coordinator for East Bay Regional Parks, managing the Parking at Day Camp and Adventure Crew programs. In addition, Morgan began her career in the Park District in 2008 as a docent for Tilden Nature Area. In 2008, she joined the ISA, or Interpretive Student Aid staff, while working on her Ranger / Naturalist certificate. Her many roles have also included docent at the Regional Parks Botanic Garden, Naturalist Aid, as well as recreation leader, I want to say that's a 5, for the NPRS award-winning co-op program. Okay, so I'm going to go ahead and spotlight Morgan. You look fabulous today. Go ahead and give you a spotlight for everyone, and you can go ahead and get started.
All right, thank you, Melissa. And welcome everyone to the culinary portion of today's program. Again, my name is Morgan, and I have been participating in this fabulous event, I believe, since its inception. And generally, I've done something along the lines of boletes or chanterelle, something that I've been very familiar with. But since it's a new year, it's 2021, and I'm trying to try new things and put myself out there, I decided that I was going to focus on a mushroom that I am not overly familiar with, and that is the Matsutake. So this is an example, a specimen of an American Matsutake. It is the white Matsutake. It's Tricholoma magnivelare, and it is related to the brown Matsutake that grows in Japan, but this one is, you would think this was a brown Matsutake just because it's not a very white specimen, but it is a gilled mushroom, which is a mushroom that I tended to shy away from when I first started mushroom hunting on my own. So when we talk about choice edibles, a lot of the choice edibles tend to have what we call reduced gills or pores or something that's not like a true gill, but this is a true gilled mushroom. I'm not really sure if you can see the gills on this mushroom, but when you are looking for this mushroom out and about in the Bay Area, you don't want to mistake this mushroom for an amanita. So when you do forage for this mushroom and you go to harvest it, you're going to want to take out the entire specimen. You're not going to want to take a knife and cut it in the ground. You want to make sure that you get the entire specimen out so that you can see that the bottom of this mushroom does not have that egg sac that would tell you that this is in the Amanita species, which tend to have toxic, deadly mushrooms. So anyway, here we have our Matsutake, and some other characteristics of this mushroom are that it has kind of a cottony top to the cap, and the stipe or the stem tends to kind of go at an angle. So you can see maybe on this specimen where it's kind of coming down, it's not growing straight out of the ground. So that is a good key factor in identifying this mushroom. Also, the appeal for this mushroom, unlike most other choice edibles, is not so much in the taste, as I've discovered. It's more about the aroma. So when you're looking for this mushroom in the field, you can take your knife and scrape the stem or the stipe and smell. And if you've read David Aurora's book that Trent just talked about, all the mushrooms demystified, He likens the smell of this mushroom to gym socks and red hots. So it's kind of an odd combination of things when you're talking about wanting to eat things, but that's part of its charm. So this mushroom is a type of mushroom where we're not gonna wanna do a long saute method to kind of draw out flavors. We are gonna wanna try to preserve that aroma. So ways that we would present this at the table would be steaming it or cooking it over a pot of rice. You can also make what's called a chawanmushi or a custard, and that's what we're gonna make today. So we are gonna use a recipe from this book here today called The Wild Table. Its authors are Connie Green and Sarah Scott. And the recipe is actually authored by a man named Hirosone. And Hirosone and his wife, Lissu Dumane, they had a restaurant up in Yountville called Terra. They actually met down in LA at a restaurant called Spago, and they moved up here and started their restaurant probably about 30 years ago. They also had a restaurant in San Francisco called Ame. He was a 1 Michelin star chef, and this is his recipe for spotlighting the characteristics of the Matsutake. So I'm gonna go over some of the ingredients that are gonna go in our recipe today, and that is a dashi broth. Now, I made this myself a couple of days ago, and the ingredients for this are not going to be staples that you have in your pantry necessarily, but we have a lot of great markets around in the Bay Area. I got all of my ingredients for this recipe from Tokyo Fish Market down on San Pablo in Berkeley. So the two things that are in this broth are kombu, which is a type of seaweed, and the bonito flakes, which are from the tuna fish. So it takes a couple of hours to create this broth. We're not gonna make it together today because we just don't have time. But you're gonna basically bring a pot of water to boil with the kombu, let it steep for about 20 minutes, then take that out, add the bonito flakes to give it more seasoning, and then strain it out, and you want a nice clear liquid as you see here in this jar. Now, if you wanted to make this recipe for someone who did not eat fish, you can also go and buy all kinds of instant dashi broths. This one is a shiitake-based one, and so it does not have any fish in it. So the dashi broth is one of our main ingredients. We have mirin, which is a sweet rice wine. This one is organic. It's got sweet rice, distilled rice wine, and sugar and salt. Has kind of a viscous texture. And then we have tamari. The recipe actually calls for white soy sauce. I couldn't find it, so we're just gonna use the tamari and add some water to kind of thin it out. Then we're gonna have some eggs that we're gonna beat up in a second. And what else is in our recipe today? Then whatever you want to add as far as flavoring. So we're going to use our Matsutake that I actually did not forage for myself. I went and bought these at Monterey Market, which is a great place to go and buy all kinds of interesting choice edibles. Luckily, they were still in season and they had them there. And then we're going to use some asparagus. Now, you can put all kinds of stuff in this chawanmushi, this custard that you want. You can put shrimp. One of the recipes I saw online had crab meat in it. It looked really good, but we're gonna keep it simple today and just go with the mushroom and the asparagus. So let's see, we are gonna take our specimen, and again, this one's been in the fridge a couple days, so it's not white. You would almost think this was a brown Matsutake, but if you have a mushroom brush, you can kind of clean off whatever lingering dirt is on the stipe or the cap. Or you can, if it's very dirty, you can take your knife and go ahead and just scrape off all of that stuff. You don't want to get any sort of gritty texture into your custard. So I'm just going to scrape this real quick. And I found Matsutake 3 places in the Bay Area, at times when I wasn't actually looking for them. And they tend to favor madrones, manzanita, pines. Obviously, the name in Japanese for this mushroom is Matsutake. Matsu means pine, take means mushroom. And so obviously it is going to associate with pines. And tan bark oaks, if I didn't say that already. And it prefers sandy soils, which is why you want to do a good job cleaning your mushroom, because you don't want to end up with sandy grit in your very delicate, savory custard. So I'm just going to dump this in the garbage here. Excuse me for a second. And then we're going to go ahead and slice this nice and thin. Again, we're not going to even cook this at all. We're going to put it straight into the custard because we want to preserve as much of that aroma as possible. You can see the flesh on this is nice and white. And other things about Matsutake, fun facts, back in the 1990s, one specimen that was commercially foraged for in the Pacific Northwest fetched over $400. So in Japan, this is a highly prized mushroom. Again, this is not the identical species that grows in Japan that associates with red pines. And those forests right now are compromised. They're have some sort of disease. And so those forests actually are disappearing. And so the price for the Matsutake is probably even greater. So again, this is a commercially foraged mushroom. It does not take well to being grown in a lab. So if you want it, you have to either go looking for it yourself or purchase it from a market like Monterey Market, where they would have, bought it from a commercial collector. So here are our mushrooms. They're ready to go. We also want to use some asparagus. So ahead of time, I took our asparagus, our baby asparagus, and I blanched it. So we can put those in our cups. And let's see, we're gonna go ahead and take out 3 eggs. If you start with things room temperature, it's gonna make it a faster process when your custards go into the oven. Going to lightly whisk these eggs because we don't want to incorporate a lot of air into the custard, because you don't want to have any air bubbles or anything that would mar the surface. Again, it's a good visual presentation to have a nice slick surface to your custard. So again, I'm just kind of gently whisking these. I'm not incorporating a lot of air into them. Then we're going to take 2 cups of our dashi broth that we made ahead of time. You can either go through the step of making it yourself with those 2 ingredients, or use a instant dashi broth, like I said before. Okay, so we're going to measure out 2 cups of our dashi broth. And you want to make sure if there's any sort of grit at the bottom, you want to make sure that doesn't get into your custard. I'm going to stir that up a little bit. We're also going to add the mirin, which is again is our sweetened rice wine. And it calls for 3 tablespoons. If you can see how viscous this is. Oops, a little extra. And then our tamari. So the tamari is not made with a wheat product. So soy sauce, people may just assume that is a soy-based product, and it does have soybeans in it, but it also has wheat. So for your friends who are having issues with wheat, tamar is a good choice. So we're going to stir that up. And there's a lot of actual liquid to the eggs, so it's not going to be a very firm custard. In fact, it's going to be very delicate, and when it comes out of the oven, you might think that it's not done, but it's just going to be a very light kind of jiggly texture to it. So that is the base for our custard. I'll just leave it there where you can take a look at it. And now we're going to talk about the actual vessel for your chawan mushi. So chawan mushi, again, is a Japanese term. Chawan means teacup, and mushi means steam. So this is an actual vessel that is designed for this particular application. It has a nice little lid to help it steam when it's in the water bath. If you don't have this exact type of container, you can always use a teacup. This one has a strainer, but we can use this and then cover it with this lid. If you don't even have this, you can just wrap the surface here with some saran wrap before you put it in the water bath. So very simple. We are going to take our fillings and put them into our cups. And again, you can put anything you want in here, but we want to really showcase our Matsutake. So we have our cups, and then we're going to actually strain this before we add it to the cups. So you want a fine mesh strainer. See here my fine mesh strainer. I'm going to do it straight into this measuring cup because then I can pour it easily without making a huge mess, which I want to do most of the time. I'm going to strain it through the strainer, and this is just getting out any bits of the egg, the chalaze, or whatever that you don't want in your custard. You want a nice, even texture to your custard. So I'm going to strain that out. And you'll see there'll be some stuff left behind in here. That's probably plenty. So then we're going to take our custard. We're going to pour it into our cups. We're not going to fill it all the way, just most of the way. And then we're going to put on the lids. Now you want to find a pan that is going to allow you to bring the water about 2/3 of the way up your custard cups. So this is a good choice. And then we would have some boiling water. You can use your electric kettle. You're going to pour that in 2/3 of the way. And just note that if you don't have this type of vessel and you wanted to use, say, a mason jar, something like this, if this were cold and your water were really hot, your glass jar would most likely shatter. So you want to use something that's ceramic that isn't going to break on you. And then we're going to cover it with foil. And we would have preheated our oven to 325 degrees. And then you're going to put this in the oven for 40 minutes at 325 degrees. And while that's cooking, you're going to want to make your glaze. Now, this is just for your personal preference if you want to make it look extra fancy. We're going to turn up our burner here. And the glaze has more dashi. So a 1/2 cup of dashi. I'm not sure I have a 1/2 cup left. But you can always use water. It's not that particular. Just Let's add a little more water to this. Also in our recipe, we have 1 teaspoon of soy sauce. We're going to use our tamari. And let's see, more mirin, another teaspoon. Whoops, that comes out fast. And a pinch of kosher salt. All right. And you're going to bring this, I believe, to a boil. Turn it down. Let it simmer for 2 minutes, and then we're going to add in some cornstarch. So while this is coming up to temperature, we'll talk a little bit more about some of these ingredients. So the bonita flakes, you can buy these at any Asian market. This is a great thing if you have a cat. They go crazy over this. If you go to the pet food store, you can buy a very small package of this for a lot of money, but you can buy it at Costco in a big bag and feed it to your cat when he's looking for a snack. The dashi is a lot of these products that are pre-packaged have MSG in them, so you have to look at the labels very closely if you are trying to avoid that, but this is a nice substitute if you have friends who don't eat meat. So this is coming up to a nice boil here. And so you'll be doing this part, the sauce, while your custards are cooking in the oven. I believe we also put some Matsutake into this glaze as well, because you can never have too much Matsutake. And so in the meantime, we're going to check on our custards while that's working. So we're going to pull these out of our imaginary oven. Uncover the foil, make sure not to burn yourself with the steam. And then we're going to take a look at these. Now, it's going to have a very loose kind of jiggly texture. I'm not sure if you can see that, but don't be alarmed and think that it's not set. It actually is. You can test it with a toothpick or with your knife to see if the custard actually cooked all the way through. When it comes out clean like that, it's actually, this was properly cooked. These are done. So I'm going to pull these out. And again, if you were going to present this at the table, you would leave the lid on and open it up so everyone can can smell the aroma. So, like so. Yeah, and I'm getting the smell of a little bit of the red hots and the sweat socks. So, perfect. And our glaze is just about ready for the cornstarch. And let's see how much cornstarch. We have 1 teaspoon of cornstarch. Now you're going to want to pull this off of the stove before you add the cornstarch. Otherwise, it's going to thicken up a little too quickly. Just know that from experience. So let's turn this off. 1 teaspoon cornstarch. Put this back on just for a minute to thicken up a little bit more. And we're gonna get our garnish ready as well. So we have lemon zest. The recipe actually called for uzu, which is a special citrus that you might find this time of year at a place like Monterey Market. I couldn't find it this time around, so we're just having some Meyer lemon zest. This is mainly just for aesthetic appeal, and some chives for something green. Cut them on a bias. Have those ready to go. And I turn that off and we'll sample this one. Gonna put the glaze on the top. You want to put the glaze on the top, ideally without rupturing the surface, and present it with a couple bits of the Matsutake. Now we're gonna put on our lemon zest and our chives. And voila. This is our chawanmushi, meant to showcase our very delicate Matsutake. So I'm gonna give it a taste here, see how it goes. Delicious. It's very nice with the broth, the dashi broth that I made, and the Matsutake. I have to say, when I first tried Matsutake, I didn't really like the texture of it. I thought it was a little too firm. And I realized while making this preparation that I just had the wrong method. I had overcooked the mushrooms. So you want something where it's just a very delicate presentation to showcase the texture and the scent of this mushroom. Now, another way to present this mushroom, we're not gonna actually do today, but something to keep in mind if you like to go car camping and you're mushroom hunting and you're out in the woods somewhere and you want an easy preparation that's not gonna take obviously all of this behind the scenes preparation, is foil wrap Matsutake with the soy and the ginger. So I'll just show you real quick. We would take some foil and we would brush it with oil. We would take our Matsutake slices and we would mix them with what Hirosone calls his mother sauce. This is akin to a teriyaki sauce, but it has the addition of sake and mirin in it as well. So ginger, garlic, soy, sugar, obviously, and then the addition of sake and mirin. Stir that up, get it good and coated. It's a very simple preparation. I'm gonna put those mushrooms in the foil. He has it obviously a little bit nicely presented there. Make a little packet. And then you want to throw this on some low coals, or even right on the grill of your campsite. And so again, when you open this up, you're going to get this amazing aroma. So that's it for our preparation. I'm wondering if we have any questions.
Yes, we have a couple of questions.
I'll take them while I eat this since I didn't have lunch.
Okay, so Mark L was asking, well, he kind of said, I'm taking notes, but missing some steps here and there. Will Morgan publish the recipe in writing? Are you going to be sharing the recipe with us at some point?
Yes, hopefully we will be able to put that in the chat later. But just know that you can always Google chawanmushi. This is a very common recipe in Japan to showcase a nice custard. So You can always just Google that. You can Google Hirosone's chawanmushi and it will come up, but we'll put it in the chat.
Great.
Is there something specific, like a specific step that was not clear?
I'm not sure. Just
Okay.
said that they're missing some steps.
Okay.
As they might go ahead and comment again. Nora asks, please remind me, why don't we wash mushrooms with water when prepping?
Because the mushroom is like a sponge, so it's gonna like soak up all that liquid. And sometimes when I'm foraging for mushrooms and they're super dirty, when I get them home, I will quickly rinse them under water, but I always will do a dry saute method with those, which entails a very hot pan and no oil to begin with. And you put the mushrooms in that pan and the water is gonna, you know, cool off and you can dump that off and save it and use it, or let the mushrooms reabsorb it. But you don't want your mushrooms super soggy. It gives it a really bad texture. And because we don't want to cook this mushroom too much, because we want to preserve the fragrance, we definitely don't want to get it waterlogged. So this one, it grows in sandy soil mainly, so it's pretty easy to clean with a brush. Obviously, you don't need this fancy brush. You can use a clean toothbrush or something, and you can always use a paring knife to scrape off the top or clean off the stipe. It's a very simple thing to do. But yeah, mushrooms are like a sponge and they'll just absorb all that liquid and then it ruins the texture of the mushroom.
Great. There was a question about the oven temperature that just came up.
So 325 degrees now, you know, that's what the recipe says, but you need to know that your oven is actually calibrated to that. It's easy to buy just an oven thermometer and put it in there, but it's a very low temperature and a slow kind of bake in the oven.
What is your favorite mushroom to cook with?
Oh, my favorite choice edible is the black chanterelle. So I love to go foraging for that mushroom. I love the kind of earthy but kind of apricot-y aroma of that mushroom. I love to make simple preparation with a light cream sauce. A lot of mushrooms are choice edibles pair very well with a cream sauce, whereas this Matsutake is not gonna go really well with that type of preparation. I do have a friend named Keiko, who is a Japanese American woman, and she's married to an Italian American. And she actually takes this mushroom and does a very long, low, slow, almost like a caramelization of this mushroom. I haven't tried that preparation, but again, something where it kind of showcases the smell. But for me, the black chanterelle, or just recently I found a king bolete, and I did a very simple brushing with the olive oil and on the grill. Morels too, also stuffed with bacon. There's a great recipe in this book for bacon stuffed or goat cheese stuffed morels wrapped in bacon. You put those on the grill. Be a nice thing to do when you're in your campsite as well. But yeah, black chanterelles are my all-time favorite choice edible.
Another question. Many folks out there are somewhat horrified at the thought of cooking with wild mushrooms. What do you say to them?
I have my degree in the culinary arts, and when I was in cooking school, I had to do an externship for 2 months in the restaurant of my choice. And so I went right for the top, and I chose Chez Panisse. And I was there for 2 months in the wintertime, and being the lowest person on the totem pole, I was tasked with cleaning sheet pans brimming with wild mushrooms. So black chanterelles, hedgehogs, golden chanterelles, you name it. And so I had never really thought about wild mushrooms or looked for them before, but after 2 months of handling them and cleaning them, I was like, I know what these things look like. I'm just gonna go out there and look on my own. And I got a guidebook. I got the David Aurora's little hip pocket guide and went out foraging on my own. And, you know, I stumbled here and there and picked some things that I didn't know what they were. But for me, at least coming from a novice standpoint and being self-taught, I always veered away from the gilled mushrooms. So again, we talk about true gills and how a lot of the choice edibles, they don't have true gills. They either have ridges, so they're kind of primitive gills. The chanterelles have a primitive gill. The hedgehogs have what we call teeth that kind of stick down. The boletes have pores, and it's all the same purpose is to release the spores, right? But when we're talking about gilled mushrooms, now we're getting into a category that may have something deadly poisonous. So I always veered away from those. So you want to stick with something that's very easily to recognize, like the golden chanterelle. Collect many specimens. Go to a place like Monterey Market and just go up and down the aisle, look at all the mushrooms. You just, get yourself accustomed to what they look like. Go out with people who do forage and who know what things are, because there are obviously lookalikes. Even this one has some sort of lookalike. I can't remember the name of it right now. Maybe Christian could tell us what is mimicking this mushroom. But you always want to have verification from someone who actually knows what they're doing. And always start with just a very small amount. You can take a little taste of the cap. If it's bitter, maybe you don't want to eat that. But yeah, going out with people that do know what the mushrooms are and really just getting to know what they look like, spending some time with them.
All right. And just as a reminder, in the East Bay Regional Parks, unfortunately, we're not supposed to be collecting mushrooms. However, you can go to places like Salt Point State Park and most national forests. Just make sure you check before you go out exploring and hunting. Morgan, there was also a question of, if you're eating these mushrooms raw, should you still wash them, or is it okay to wash them then if you're eating them raw?
There's a great preparation that came out of Chez Panisse for bolete mushrooms, where it's you got to find a nice specimen that doesn't have any maggots. So you want to get that mushroom when it's really young. You slice it really thinly. You put it on the plate with some thinly shaved fennel, Meyer lemon juice, olive oil, salt and pepper. Super simple. Obviously, the Chez Panisse way really showcases the special flavors of the mushroom. But they're not going to wash that mushroom. They're going to take that brush and they're going to clean it right. Like I would never really recommend washing mushrooms unless they're really dirty. If they're really dirty, you got to wash them, then you're not going to want to eat them. So yeah, sticking away from washing, cleaning off all the dirt, again, just using your knife or your brush. Like you saw, I barely brushed this off and I'm eating it and there's no dirt, but you just need to make sure that, yeah, you clean it properly.
Great. Can you mention the name of the book again?
Oh, yeah.
Folks? Yeah.
Yeah. This is called The Wild Table. It has all kinds of great recipes. There's also a really good recipe in here for porcini dusted ribeye. So you can take your porcinis or your boletes or your ceps or whatever you want to call them. Even the suillas, it's got a slimy texture, but if you dry that mushroom and you grind it up in a very clean coffee grinder, so you can have a coffee grinder that you have just dedicated to your herbs or your spices, as opposed to one you use for your coffee, or you can clean it out by using some moist breadcrumbs. Just puree it, and then dump it out, and then your coffee grinder is clean. But you can make a nice powder, and you can use that to season things. You can make your own salts, but this recipe in this book has the porcini dust that you put on the ribeye before you grill it, and it's delicious.
Fantastic. Morgan, is there anything else that you would like to share? I've kind of run the gamut on all of our questions.
Yeah, so as far as where to forage, so Point Reyes, Salt Point, Jefferson Demonstration Forest, I believe, but also make friends with people who have open oak woodland forests so that you can go forage on their properties.
All right, one final question from Alex. Maureen, do you have any suggestions for wild mushrooms that make good mushroom jerky?
Oh, well, I have bought shiitake mushroom kind of jerky. I think that would be a really good choice. You could experiment, you know, it's just when you're drying them, you want to make sure that those specimens that you're starting off with are really fresh. Otherwise, they're kind of gonna get kind of mucky. Like you might want to use a dehydrator that you can control the temperature on, or do it in your oven. Yeah, shiitake. I would start with that.
Fantastic. All right, everyone. Morgan, thank you so much for taking us this culinary journey. It was awesome. It's also so great to see other colleagues' skill sets. Like, this is fantastic. I loved it.
Thank you.
Thank you so much. And we're going to go ahead. I think we're going to go ahead and transition into another activity. Thank you.
Thank you.
All right. Thanks, Melissa, for hosting Morgan there. And thanks, Morgan. And also mad thanks to Naturalist Anthony Fisher, who's running all of our audio video behind the scenes there. He's that mask face peeking in that you may or may not be able to see. Folks, we're going to take a short break. Up next, we have got Mushrooms of Lake Merritt with Damon Tighe. That's going to start at 12:45. So we'll be out for just a little under 20 minutes. In the meantime, please enjoy the photography of local photographer and mushroom enthusiast Anna Towers. You can find her on Instagram at The Friendly Fungus. Look her up. We're going to play a slideshow of her photography while we wait for 12:45 to roll around. Up next, Mushrooms of Lake Merritt.
trees have gone home. And Olympia rides a crimson tide on the river alone. The hemlocks quiver before she speaks. The trees have gone home. And she feeds all of the mountain creeks on the river alone. And we will kill Olympia. We will kill Olympia. Fair Olympia, the scorned loggers bride, the trees have gone home. Got both the captain and the long logger's eye on the river alone. Both men came to her bedside lamp, the trees have gone home. But she would not take any man in camp on the river alone. And we will kill Olympia. We will kill Olympia. In her woodland home, as the trees have gone home, and wash her body down in the melting snow on the river alone. And brave Olympia, the scorned logger's wife, ere the trees have gone home, stood her ground to protect her life on the river alone. And hunt her down, we'll kill Olympia and her sweet lips will kiss the ground. Hunt her down, we'll kill Olympia and her sweet lips will kiss the ground.
All right, everyone, welcome back to the 2021 Tilden Fungus Fair. I'm your host, Trent Pearce, here at the Tilden Nature Area in Berkeley, California. That last musical interlude was brought to you by a good friend of mine, Dan Kirkwood. He's up in Juneau, Alaska. You can check him out on Bandcamp or Spotify, Dan Kirkwood. I got a little tired of the trance music that I originally put with that slideshow video, so thought I'd give everyone a break. Well, it's my pleasure to introduce our next guest speaker, Damon Tighe. How's it going, Damon?
Great, Trent. How you doing?
Fantastic. I was thinking we could ham it up for just a minute before we tear into your introduction and presentation. Can you remind me how long we've known each other?
We met through a guy that I met on the internet. His name was James Frank, and I think that happened in 2008, maybe.
Wow, that's a pretty solid run. I thought we met through the old, now mostly defunct meetup group, Nerds for Nature, but maybe that was just the first time we hung out in person.
I think that was the first time we hung out in person. Yeah.
Yeah. So this was Nerds for Nature was a literal meetup group on meetup.com, and it was designed to get nerdy people together to kind of use their collective brain power for environmental good. I think Damon and I pretty quickly realized that our collective brain power revolved around fungi and photography and adventures. And yeah, we started hanging out and going out, picking mushrooms and comparing photography notes. And it's just been nerdy bliss ever since.
Pretty much. Yeah. It usually pivots around the winter time and those mushrooms come up, but occasionally also get in a couple summer hikes and things like that. So it's been great.
Yeah. Well, Damon's a pretty busy guy, so I feel really fortunate to have him here today. I know he's been doing all kinds of presentations already this week. So yeah, thanks for coming, Damon. I'm going to
That
tell
is-
a little bit about you now and then let you get into
Uh-oh.
the presentation. Yes. Damon was born in Klamath Falls, Oregon, and raised in Calaveras County, California. He attended St. Mary's College of Moraga, where he worked on local newspapers while earning a biochemistry degree. He then dabbled in teaching high school in Portland. I don't think I knew that about you. Before moving back to the Bay Area to work on the Human Genome Project at the National Labs Joint Genome Institute. He currently works for Biorad Laboratories, designing curriculum and training educators in biotechnology. And he's very well versed in local fungi, plants, and animals of Lake Merritt. Please make virtually welcome Damon Tighe.
All right, fantastic. Good to see everybody here today. I'm going to go ahead and share my screen, and I'm going to use a slide deck to go through the presentation today. I'm also just going to drop a link for that slide deck in the chat. And so that way anybody can get back to this and be able to see that slide deck. You're comparing notes against mushrooms you might have locally. So one of the cool things about Lake Merritt is that, hey, it's in the middle of a freaking city and there's a ton of things that go on there. So it's actually a really good place to understand urban mushrooms in general in California. So a lot of the stuff you see here might translate to stuff literally in your neighborhood, which is kind of cool. So with that being said, let me share my screen and I'm going to jump into that slide deck. Make sure this works. Trent yelled for some reason. You cannot see the, the presentation.
That looks good on our end, Damon.
All right, see a bunch of guys jumping in there. Okay, so let me give you the general kind of arc of what I'm gonna be doing. I'm gonna go through a little bit of mushroom basics with you because I think it's always good to make sure you've got that foundation before moving on to talk about specifics. I'm also gonna talk about a platform called iNaturalist that I use obsessively, and some of you have been using this weekend. And why iNaturalist? Well, it was built locally out of a guy that lives here in Oakland, and it's just a great tool, like modern day notebook, to keep track of the things that you've seen, which allows you to learn, and you get to interface with a group of people that will help you identify things. And then we'll basically do our local species tour of Lake Merritt, and I guarantee you some of you guys will be blown away by what you can find just in an urban park. Remember, I've been doing this, looking at critters around Oakland or in Lake Merritt specifically, since about 2004, since I moved here from Montana. So this is, the culmination of about a decade worth of poking and nudging critters. Big picture, fungi. Compared to other critters in the basically tree of life hierarchy, they're pretty darn close to us if you're talking about where they used to be placed, which was next to plants. If we think about fungi in their own little portion of the branch of life, most of the things we think about when we think about mushrooms occur at just one kind of far end of that branch of the taxonomic kingdom, fungi. And that tends to be the basidiomycetes. These are like the big gilled things that we see and we think about and eat. And some of the cup mushrooms, the ascomycetes, and the hallmark one for this, of course, is the morel. These are the two that produce these large enough fruiting bodies that we can kind of find them as we're walking around through Lake Merritt. There's great simple keys to these online. There's a place called mycoweb.com/CAF that has a great kind of really user-friendly key for somebody who's just getting started. So for the basidiomycetes, it breaks it down into kind of general shapes and forms. Does it have gills? Does it have pores? If I poke it, is it jelly feeling? Things like that. They've got a similar thing for ascomycetes. So this is also, if you don't already have some of those great guidebooks that Trent talked about earlier today, this is another great kind of resource that's free that's out there, mycoweb.com/CAF. Also, one thing that's important to think about when we think about mushrooms is that the mushroom is literally just a very small portion of the life cycle of that species that we're talking about. It's basically kind of the sexual business end of the mushroom life cycle. It comes out there in order to get spores out into the world. Those spores end up places that they like. They can then grow to be hyphae. They can find other hyphae. They can have hyphal love, form little basically hyphal knots. And then when conditions are right, so if we're in California, this tends to be our winter when we get rain. Oh God, please let there come more rain. These little hyphal knots will develop into primordia, burst through the ground or their substrate, and all of a sudden you've got mushrooms out and abound. For identifying those basidiomycetes, especially the gilled stuff, looking at how the gills attach to the stem is really important. So here's like a basic key to kind of think about. Also, the cap shapes can be very helpful. Are they flat? Are they kind of dome shaped? Do they have a divot in the center? All of those little attributes are things we want to start to be able to kind of see and think about when we find mushrooms around us, because these are the factors that allow us to kind of start keying them out in the books or these online resources that are out there. And this is how I started. I think I started, well, I actually started because my girlfriend at the time thought I was going to kill us, and so got me David Aurora's Everything the Rain Promises and More, which has keys like this in there. I started going through those, and that's basically you just kind of slowly build this language of how to identify these things. And some of the features like this are really important. You can also do spore printing of things that you find, and this just means cut off basically the cap of the mushroom, place it on, a dark surface or a white surface, depending upon which color you think the spore print might be, or hedge that bet like I do and place it on like a piece of glass. That way I can move it to light or dark places, and that way I can easily see what color are the spores that are falling out of the mushroom. Or if you want to go super nerdy, this is like the next level step, is you get yourself a microscope and you start looking at those microscopic features. So some of the key microscopic features are looking at how spores are shaped, chemical reactions they might have, what their sizes are, things like that. So for the ascomycetes, those cup fungi like morels and things like that, you could also look at the ascus, basically the long sac that all of those spores get packed into and then explode out of occasionally. Or for the basidiomycetes, those gilled guys, you could be looking at the spores and the basidia, the little club-shaped cells that this group is named after that the spores basically bud off of. And the full end of the nerd spectrum is you could actually take some of those mushrooms you find, extract DNA from them, and sequence that DNA and use genetic data also to help you figure out what those organisms are. And there's a handful of people doing this in the Bay Area, literally in their homes. Like I was doing this in my bathtub for a while, but there's 3 or 4 people for sure, like even just in Oakland, that are doing this. This is something that is slowly becoming more and more accessible to people, which is great because it's another lens for us to see these organisms through. There's the guides out there that Trent had talked about earlier. These are my 3 favorites that I go to all the time. I really love the way certain ones of them are laid out. And then others have, larger amounts of content, or some are just fun reading. All of the Rain Promises and More, I think, is the only guidebook I ever read, like cover to cover, because it's not just a guidebook. There's a bunch of short stories in there and things like that. But the other learning tool that's really important and really got me to where I'm at now, where I'd say like I'm a high-level amateur at identifying mushrooms, is using this platform called iNaturalist. And what this is, it's basically a phone app that you can get for your cell phone. You can take a picture of any organism, any place in the world, upload it to a global database, and people there on this platform will help you identify it. It also has an artificial intelligence now built in. They can basically look at your photographs, compare it against 4 billion or whatever observations iNat has, and give you some best guesses at what it thinks that might be. For mushrooms, for a long time, they were not very good, but they're slowly getting better as more and more people contribute information to the platform. And so I'd say for a lot of the most commonly found mushroom genera in California, iNaturalist will actually usually give that as your top guess, which is pretty amazing. And then the beauty is there's real people there. There's really great mycologists that go on there and help identify stuff. So any of you guys that were involved in the MycoBlitz this week, you'll start to see that stuff you've uploaded is being basically kind of curated, talked about on there. And people are saying, hey, I think this is actually this with your data. So it is a phenomenal learning resource. And you capture all of those things that you've seen in one place. It's a digital notebook. You can go back to it and be like, I remember I've seen this before and I identified it. Oh, yep, there it was. Now I remember what it is again. So iNaturalist, because it's using visual information, it's really important to know how to present it with good visual information to get those identifications. So those attributes we talked about in the mushrooms that are helpful, those are what you want to get in your photographs. So you want to get like a full body shot of the mushroom. You want to get how those gills attach, what the shape of the cap is, maybe what the bottom of the mushroom looks like. And if possible, you could stage those all together in one photograph. So that way, when you upload it, somebody like myself that's going through and helping identify and stuff, I can do one look at one photograph and be like, oh yeah, I know what that is. You can also add multiple photographs there. So if you can't figure out how to get them all in one photograph, you can just upload 4 or 5 photographs that get you everything that can communicate what this mushroom looks like, where it was, things like that, so that people can identify it. The beauty is, because you're using it with your phone, if you give it basically access to your GPS location, it'll also grab a GPS point for that. So when you upload it, people know where you saw that. For those of you guys that are worried about giving away like your favorite chanterelle patches and things like that, you can also just obscure the data too. And so that way people know, hey, this is happening somewhere in this 50-mile radius, but I can't tell exactly where it is. And so you can also hide data. And for any kind of critically endangered species, I think it's red list species, iNaturalist automatically scrubs the GPS so that nobody can tell exactly where it is. Other cell phone tricks you just need to know, to be aware of. Some mushrooms are really bright. And so when you're taking a photograph, it might turn out washed out. If you hold down on your screen when you're trying to take a photograph with most cell phones, you'll see what's called an auto lock feature come on. When that auto lock feature comes on, if you then kind of scrub your finger up and down or left and right, you can actually change the exposure on your photograph. So all of a sudden that chanterelle goes from being a blown out golden blob to something where I can see those primitive gills on it. Also, a lot of mushrooms can be relatively small, and so you might want tools that can get you up close. This little tool right here runs me about $1 on Amazon, maybe $2-3 on the shipping. This just clips over any cell phone. I have got a case on mine, and now I can get really close to stuff and take pictures of really tiny mushrooms. So for example, that mushroom on the left there, it is sitting next to my ring. So now you have an idea of how small that is and how much closer that little tiny cheap macro lens can kind of get you there. It's a great tool, especially for getting access to those smaller organisms. I won't go into the details of uploading iNaturalist. It's pretty straightforward, and it's different for the iPhone and for the Android for the mechanisms. Basically, you add your photos, you give a best guess at what you think that thing is, and then hit upload, up it goes to the cloud, people can start getting access to it. You can also data mine off iNaturalist. So I could go onto my phone, I could turn on the Explore button, and I could type in at the top, I want to search just for mushrooms, and it'll show me all the mushrooms around me that other people have uploaded. And so I can go, oh, that's where I want to go look. That's where I'd like to go see stuff. So know that you can kind of data mine on there. You can data mine really well on the website called iNaturalist.org, and you can do really deep dives there. So here is basically a data dive into Lake Merritt. So right now at Lake Merritt for fungi, we've got over 170 species documented. There's been over 1,000 observations. And so I'm going to take you through some of those fun ones. The first one that catches people's eye whenever they go into urban landscaping, I'd say in the Bay Area, is this bugger here, Clathrus rubor. It's A, because it's big. It's like the size of a softball. It's bright pink, and it smells like a rotting corpse. So what's not to love? What's not to miss? It has this kind of eau de death effect, which attracts flies to it. So the flies actually will fly into the cage, and you can see those little kind of like brown globular sticky things inside. That actually has the spore material on it, and that can stick to the flies, and the flies fly off someplace else and they spread the spores. This typically is found in places where you find lots of wood chips. It's one that degrades wood chips, so it loves to be in landscaping. If you can find landscaping that's getting watered all year round, pretty high chance that this is showing up at some point. These guys tend to show up kind of at the end of summer, and then will kind of progress all the way through into December some years. They are edible. Some cultures do eat the young eggs from these. But you want to get them very, very young because they get that eau de death flavor as well as they start to mature. I've only had a couple young ones, and I made one mistake on getting an olderish one time. Never again, never again. Ophilotus olivicens, the Western jack-o'-lantern, is a really cool mushroom that you can find in urban habitats. I find it at Lake Merritt all the time. In fact, some guy, El Marco on iNaturalist, found this giant cluster of them at Lake Merritt. I was like, oh, I can't believe I missed that. These are bioluminescent. They glow at night. How cool is that? They also can be used to make dyes with. So a lot of people that enjoy making fungal-based dyes, this is one of the coveted mushrooms because it produces really nice kind of purplish pigments when you dye with it. To see the bioluminescence, It's not as super bright as some of the tropical bioluminescent species, but if you go out at night and you kind of let your eyes adjust and find one of these larger open specimens, you will be able to see like a faint glow. If you've got a DSLR camera, the best thing you can do is basically set that up for a long exposure, like 30 seconds at your highest ISO, do a long exposure of it, and you'll actually be able to tell if those gills are glowing pretty brightly. And you know what? The specimens range a lot. So I've found really nice ones I thought would glow that were doing nothing. And I've found haggard ones like this one at Lake Merritt. I was like, that's probably not going to glow. And sure enough, like I was walking home with it and I was like, I can actually see it glowing while I'm walking home in city lights. Pretty amazing. The other one at Lake Merritt that is very well notarized in California is one of our best documented invasive species from Europe, Amanita phalloides, the death cap. This mushroom is responsible for the most deaths, mushroom poisonings in California. It associates with Quercus agrifolia or coast live oak, and so we'll find that on the oak trees around Lake Merritt. And at Lake Merritt, you actually get to see probably how it ended up here. You will also see it associating with the cork oak at Lake Merritt occasionally. The way to be able to recognize this, it's the Amanita family, which has these big white gills, and they emerge from kind of a little sac in the ground, a vulva, an egg-like shape. The cap color on the ones at Lake Merritt are just like the typical shiny metallic green of death caps. In other locations, they can kind of get weathered and you don't see that as much, but pretty much all the ones I ever find at Lake Merritt just have that pale metallic green that just gives me the willies. This one, the reason that you die is it stops RNA polymerase 2 in your body, which basically means you can't translate your DNA to make proteins for it. So things just start shutting down. Your organs go through failure. It's really bad because you don't recognize it right away. It's usually about 24 hours later, you start to get symptoms. And there's only a few protocols out there right now that allow people to kind of survive this. And so this is really one to kind of be very aware of, especially for anybody that's doing foraging. The other Amanita of well-known Merritt at Lake Merritt is Amanita muscaria. This one we don't see super frequently at Lake Merritt, but in big rainy seasons, I do see it there. So this is one of the only slides where I actually stole one of my Amanita muscaria photographs from somebody from some other location for that top left one. The bottom right ones are the ones I see at Lake Merritt. They usually don't get big. They're usually only about like this tall at Lake Merritt, where other places they'll get ginormous. They hang out with the cedar trees at Lake Merritt. And so there's a handful of places where there's cedar species at Lake Merritt, and these will be up usually after that first rain in the fall, especially if it's a big dumper, you'll see them pop up. These guys everybody recognizes them also as being psychoactive. This is probably not a psychoactive mushroom that anybody would really go back to for a second experience, because it is pretty rough on the body. But that's for a whole nother talk. The other mushroom that you will see in heaps at Lake Merritt, and especially most urban environments, this is the biggest mushroom I consistently see all over Oakland, which is the honey mushroom. It's a parasitic fungi. It's known for being one of the largest individuals in the world. It has like up in Southern Oregon, I think it's taking up something like 15 square kilometers or something like that. It's enormous. It fruits usually in clusters, usually next to a tree that's on its way out, or sometimes it'll be fruiting where a tree used to be because it's still basically eating through that rootstock. And Trent just put it out there. If I'm getting close to time at some point, just jump in and break me. Agaricus xanthodermis. This is the most common mushroom you're going to find in lawns at Lake Merritt and most of California. It also is just fruits gregariously. You'll see huge arcs of it. People get really excited because it looks like the button mushroom you eat at the store. This is part of the lose your lunch bunch, which means if you were to eat this, most people will have a severe gastrointestinal response to this where things come out every orifice. So not something you want to play with. But the cool thing about this mushroom is it has a fantastic staining reaction. So if you grab it and genie in the bottle it will slowly oxidize this bright yellow color, which is one of the easiest ways to identify it. You also can break the flesh and smell it. and it smells really strongly of phenol, or just kind of think about that as like lab chemical smell. You'll kind of go, yeah, that doesn't smell like I should be eating it. They start off with kind of pink gills, and those gills will slowly kind of fade to this chocolatey brown. That chocolatey brown is the same color as the spores from these. These start going actually before the rains at Lake Merritt, just because we're watering the grass there. And they'll continue usually on until at least December. I think I just found some this week, which I felt was a little late in the season, but they're still out and about. Other agaricus that are at Lake Merritt, and this is one that I actually enjoy eating every once in a while, although big cautionary tale, eating mushrooms in an urban environment. They are picking up all kinds of stuff. They are great at scavenging stuff for trees they're with, right, and things like that. They're also really good at scavenging things like heavy metals. And so a lot of these mushrooms will concentrate heavy metals from the environment in them. And so that's why there's a big cautionary flag around eating mushrooms from an urban location like Lake Merritt. Agaricus burnidae, known as the salt agaricus mushroom, is called that because it has a salty profile to it. So if you rub it and smell it, it actually does have, it has this kind of like sea salty smell. And if you cook it, it has these very kind of like salty, nice tones to it. I really like to find this mushroom in places that aren't Lake Merritt covered in trash and consume it. It's a really beautiful mushroom. The way that you tell this quickly apart from Xanthodermis, you rub it, no yellow reaction. This guy also tends to have these kind of big kind of scales on the top. It also usually does not get out of the ground very far. It's a very kind of shrumpy mushroom, likes to hang out deep in the grass. And then if you cut it, unlike Agaricus xanthodermis, which will give you a little bit of a yellow reaction when you cut it, this one will actually start to stain red over the course of about 30 seconds or so usually. So that's how you would tell these two apart. These occur all over Lake Merritt, and I've done my bestest to make sure that that is the case. Every time I find them, I take the spores and I go throw them in other lawns in the lake area. We also have Agaricus augustus, which is a prized edible agaricus mushroom that has these almond-like scents to it when you smell it. It also has kind of this almond extract flavor to it a little bit when you're eating it, so it has this nice sweetness to it. That's why it has this kind of very regal name, the Prince. It's also called the Prince because it looks like it's wearing this very dappering outfit of these really goldenish kind of fibrils on the outside. It tends to have kind of a square complexion as it's first getting up, but then will become these giant kind of planar tops that can get to be dinner plate size occasionally at Lake Merritt. These guys like really, really thick duff that stays moist most of the year, and they'll fruit oftentimes in the warmer months. So I usually find this more towards the spring or really at the front side of fall, even before the first rains have hit. Other ones that are in the grass very commonly at Lake Merritt are the white dapperling mushroom. This is Leucoagaricus, Leucothyces. And just FYI, I might butcher the Latin names. We all butcher the Latin names at some point. But the biggest thing you'll learn from talking to any fungal people is we all kind of say things differently, but we all ultimately will know what we're talking about. And so you can always go ahead and just take a stab at those words and know that that's what most everybody is doing since Latin is a dead language. Leucoagaricus, Leucothyces, the way you tell this apart from a lot of the other white kind of agaricus looking mushrooms at Lake Merritt is that these will usually fruit in clumps, little small arcs of them. Their gills stay white their entire life. They are not, though, like an amanita, because if you dig them up, they don't have a little like egg, little vulva they're emerging from, but they will have a swollen big base to the bottom of their stipe. They'll also have a really persistent annulus or kind of ring around the stem, and that's from when the cap was attached to the stem and it opens up, that ring is what's left behind on it. These guys also have a very smooth cap on them. And if you spore print them, they're bright white. It has a really closely related mushroom, the Leucoagaricus barsii, that has a lot of those same attributes, except what it's easiest to see that's different on this one, it has a really shaggy top to it, and an annulus, the little kind of skirt around the stem, will also be very, very shaggy. This is also a mushroom I enjoy consuming, but it's definitely not one you want to get to until after multiple years of foraging and being able to easily identify this against amanitas, against other agaricus and things like that. But this is a really attractive looking mushroom because it's very sturdy, and those fibers when they first come up are really, really pretty. This comes up usually early in the fall, sometimes at the end of summer when things are still warm, and the grass at Lake Merritt is getting watered really well. Closely related in morphology, but not taxonomy, is the shaggy parasol, Chlorophyllum bruneum. We actually have a couple chlorophyllums within the Bay Area, but this is the most common one that you find within the urban setting. The easiest way to tell this one apart from the other shaggy parasols is if you look at the base of the stem, or the stipe as mycologists call it, it goes all the way down and then bam, really abruptly it goes out into this bulb. And that's very indicative of Chlorophyllum bruneum. This thing loves really thick duff. So there's a handful of locations around Lake Merritt where leaves get piled up and sticks get piled up that this thing will fruit pretty gregariously. This is also a pretty good edible mushroom. But once again, the caveat of foraging in an urban environment, these things are picking up stuff. One of the most common mushrooms that I see on iNaturalist, and it's because people see it in their lawns all the time, especially if you had, say, a tree in a lawn and it got removed, years later, these come up in giant numbers. This pale brittle stem, Saphirelia candellina, it's got a tan-colored cap. It's usually kind of frayed at the edge. And like I said, it really likes grassy habitats where there's debris from woody stuff in the past. The stems on these are also, like it says, a brittle stem. So if you kind of take it and you kind of pull a little bit, it will crack relatively easily, or it will shred out into a bunch of like little fibers all over the place. A really common, super common mushroom in every single grassland at Lake Merritt is the yellow field cap. Bulbolitis tidibans, totally butchered that one. Sorry, everybody. It is bright yellow when it first comes up. And then it kind of slowly fades to this kind of pale yellow. The gills go from being kind of a whitey orange yellow color to a deeper kind of orange color. And the crazy thing is if you spore print these, it is super rusty, which doesn't look at all like what you see on the mushroom, which is kind of cool from a spore printing point of view. But this will be in the grass during the summer, usually April through August easily. Every time the grass gets watered and we get a series of warm days, these will just shoot up all over the place. They're very ubiquitous. Alongside them is often this Conocybe alpala, the milky cone cap. And it sometimes will look very similar. The easiest way to tell it apart, though, is that this one has a much more kind of domed cap to it. And this one will show the color of the spores usually a lot faster than the field cap does. So if you flip these over, you tend to see that kind of orangey, rusty color that those spores are in the gills of this one. This is also one that fruits just like that yellow field cap all throughout the summer, especially right after grass gets watered and we get a warm day, they'll just shoot out all over the place. Especially when new grass gets put in at Lake Merritt, these things are just shooting up every day all over the place. One of the other ones that gets certain mushroom people excited when they see it come up in the grass is this, what we call mower's mushroom. And those certain types of people get excited because they think this is a psychedelic mushroom, which it is not. It looks very similar to some of the psychedelic mushrooms in that it has this kind of hygrophane cap on it, which means that it will change color depending upon moisture content in it. It can be dark brown to this caramel color. This also spore prints black, but this is very ubiquitous also during the summers of Lake Merritt. It's called the mower's mushroom mainly because this is probably how it gets propagated everywhere, is they mow it over and then just spread the spores left and right. This will also usually fruit in small clusters of like 3 to 4 in one location. Marasmus oretes is a commonly consumed mushroom known as the fairy ring mushroom that you'll also find in the grasscapes of Lake Merritt. What I like about this one is it actually changes the behavior of the grass. And so you can actually go to the location that these fruit at Lake Merritt and you will see differential growth of the grass depending upon where Marasmus oretes is growing. So you'll see like more growth in the ring area that this thing grows and then less growth of grass on the outside of it. These guys have a really flexible stem on them. They're a really kind of hardy feeling mushroom. They've got white gills. They're really small, but they have that little kind of umbo to the cap, that kind of little top on them, which makes these kind of easy to spot in that regards. They have a really nice white spore print. People eat the caps on these. The stems are a little bit too chewy for most people. by far the most common mushroom in the Bay Area that gets uploaded to iNat is, of course, the chip cherry, Lorratomyces ceres. Probably originally from New Zealand or Australia, got here on some wood scraps. It loves landscaping wood chips. And this thing will fruit all year if you just give it a little bit of water. The easy way to identify it is it's got that kind of bright red illustrious cap. And if you flip it over, a lot of times on the stem, you'll find kind of red marks going down the stipe. Questionable about edibility. David Aurora has consumed this and made some posts about it. I haven't quite taken myself there just because most of the places I find this come with other things I don't want going in my mouth. Copernellus Zeus, the mica cap, is also a mushroom you find very commonly in all urban habitats. I find it really frequently at Lake Merritt whenever they take out a tree. This will pop up in giant numbers along the edge of a stump. These are kind of a cool mushroom in that they deliquesce. And what deliquesce means is that they self-melt. They produce enzymes that then digest their own chitin. And so they kind of go from being this cute little mushroom one day to this like slimy unfurling cap the next day, which I think is a strategy for getting yourself stuck to organisms walking by and spreading your spores around. The way to differentiate this Copernellus from other ones is it has a bunch of little speckles basically on the outside of it, which you can kind of see in that middle photograph there. It looks like somebody had thrown some of those ice cream condiment tops on it, but they will kind of flock off in time. Copernopsis lagomos, the hare's foot ink cap, is another one you will commonly see in landscaping wood chips, and especially mulch. In fact, any of you guys that have planted pots at home, you probably have also seen this. The reason it's called a hare's foot is it looks like those horrible little keychain things that people used to have in the 80s that was like a rabbit's foot. It looks exactly like that when it first emerges from the ground, and then very quickly, usually within a day or two, it will shoot up and then open up and drop its spores and then be out of there. This is one that you've got probably about 24 to 48 hours to see this from when it emerges from the ground. Otherwise, it's gone. Hypholoma fasciculata, the sulfur tuft, we also have around Lake Merritt. It also likes wood chips and where previous trees were. The cool thing about this one is it is extremely UV fluorescent. So if you're one of those weirdos who walks around with a UV light at night for kicks, this is one of the first mushrooms you will find. You'll hit that with your light and it just beams back this bright green color. Not quite sure why UV fluorescence takes place in some mushrooms. It might help attract certain insects. The sulfur tuft, though, is also very easy to tell because it just has this just kind of mesmerizing yellow color to the cap. And when you flip it over, it'll have kind of a greenish hue to the gills, slightly with like a little smoky aspect to it. These will fruit really gregariously in kind of more naturalized landscapes, like in some of our parks on the outside of cities where there's larger kind of decaying bodies. The bummer about a lot of urban parks is they don't like decay, which is too bad. There was a place in, I think it was in Victoria, BC, where there's a park where they're actually doing intentional decay. They'll bring logs to rot there, and I really wish we would do that here because you'd have much more diversity. But that's why you see these fruit in really big numbers in some of the parks in the East Bay Hills or down the peninsulas, because they just are finding larger debris piles to kind of chomp their way through. Gymnopus brassicolans, the cabbage parachute, is also a ubiquitous mushroom within landscaping wood chips at Lake Merritt. The way you tell this one apart from all the other ones is it's got a super flexible stem on it. You can't even break it. I dare you, try to break it. It's really difficult. It's also black towards the bottom and then gets light right before it interfaces with those light colored gills. And the key feature is you take that mushroom and kind of smash it in your hands a bit and do this. It smells like somebody just put a cabbage fart in your hands and you just inhaled it. It has a very, very distinctive scent to it. You'll never forget it if you ever smell one. But also something that we won't usually see until usually midwinter or late winter in the landscaping wood chips around Lake Merritt. Tubarias are also everywhere. These are one of the most predominant species in wood chips. They get overlooked a lot though, because they're kind of drab, not really colorful. There are no Leratomyces series. They're not like, hey, I'm red, here I am. There's probably a bigger diversity of these than we understand in California, but right now a lot of them are getting this species name. Common name on these is the scurfy twiglet. The easiest way I find for me to identify these against other ones is when I look at the gills. A lot of times the gills are attached or just slightly decurrent, and they tend to have kind of an orangish color to them. And if I'm finding fresh samples, I can find veil remnants on the cap edge. So if that center picture there, you see those dotted white lines going around the cap, those are the old remnants of the veil. And that's a very distinctive thing that you see on tubaria as they're emerging. We also at Lake Merritt have the false turkey tail, the Sterum hirsutum, also known as the hairy curtain, because it has a bunch of hairs on it and it looks kind of like a curtain. These guys were often confused with turkey tails for people that are just beginning, because they live in the same habitat. They like to be on dead oak or dying oak, and they've got this kind of variated color pattern on the top of their shelf. However, though, if you flip them over, they have a smooth pore surface, where the turkey tail will have a white pore surface. And the pores on these are so small you can't see them with your eyes, where the turkey tail will actually have noticeable pores that you can see. The bluet is also very common at Lake Merritt in the oak duff. This is a prized edible mushroom. People say that it smells like orange juice concentrate. I'm not sure, David Aurora must have come up with that. I say fruity. It smells fruity to me. It is one of the most amazingly blue, purple hued mushrooms you'll find in the Bay Area, but you have to find fresh ones to get that color. They'll quickly fade to be kind of a brownish color in age. But one of the ones that when I first moved to Oakland, I was collecting around these at Lake Merritt, and I started thinking about what else are they picking up? Maybe I'll just eat bluets from the hills instead. Or from, Salt Point, places like that. But beautiful mushroom, one I would really encourage you to try and find in the duff just for the visual gratification of it. One of the other really prized forageable type of mushrooms that you could see at Lake Merritt is the candy cap, Lactarius rubidis. So this is in the Lactarius genus, which is a group that produces latex. So they produce milk per se. And on this one, when you cut it, it'll bleed out this latex, and it's this kind of what we call like 2%, non-fat milk look. It's not really white, it's not really clear, it's kind of in between, and it doesn't change color. If this mushroom dries down, it actually has the scent of maple syrup to it. So people actually work with this as a flavoring for a lot of desserts and sweets. I used to use this one a lot to make beers with. It's a great kind of non-fermentable sweetener in your beer. I've also made cookies with it. Really, really fun mushroom to work with. The kick though is they've got to get dry, and then they've got to not be crispy dry in order to develop that smell. So they need a little bit of moisture in order to run this reaction that develops the compound that has that maple syrup context to it. The funny thing about this mushroom is I knew it was at Lake Merritt because I could smell it. For 2 years, I could smell it, but I couldn't find a freaking mushroom to save my life. And then finally one year I went out earlier than I had smelled it and looked in that area, and I started finding them fruiting there. And I was like, finally, I know that they're there. And so the smell is really intoxicating sometimes. We have another quote unquote candy cap, the Southern candy cap, or the Rufus candy cap, also at Lake Merritt. The way you differentiate this from the other one is this one's got a lot more kind of reddy tones, rusty tones to the body of the fruiting body. If you cut the stipe on this, this one tends to have a solid stem. The other rubidis tends to be open. And this one, when you cut it, the latex is much more white than it is kind of 2% 2% milk. This one doesn't smell as strong as rubidis, but if you work with it, i.e. you dry it, let it get a little damp, re-dry it again, you can definitely build the tones up to be very, very similar. This is actually one at Lake Merritt that we sequenced as part of Barcode the Lake, a project that I'm involved in. And it was really cool because when we sequenced this mushroom, it came back to not fit anything in the databases, which was like, what? So this thing either the holotype never got sequenced, or we don't maybe have the exact right name on it. There's a lot of taxonomy work that needs to be done in California. A lot of it's going to be taking using microscopy and DNA sequence to kind of get us there. We have some rare stuff at the lake. So we have Schizophyllum amplibum, which is this thing that you usually only see in Europe because it exists on European poplar trees. But guess what? At Lake Merritt, we've got European poplar trees. And so I think I had like the second observation ever in North America, just because we have those trees there. And then once we started kind of putting the call out to people like, hey, start checking your poplar trees, we started realizing that, like, you know what, you can find it in lots of places in California, in urban parks that have those European poplar trees. It's a nice kind of small, it looks like a cup mushroom, but it is actually a Basidiomycete. It's closely related to Schizophyllum commune, which is one of the most common mushrooms across the world. Really gorgeous little guy, but really kind of hard to find unless you know how to go find poplar trees and then dig through their sticks. Another cool one you can find at Lake Merritt on debris is the cannonball fungus. And so you can see on the bottom kind of left-hand side there, this thing has a little projectile that it can actually shoot out. These things can shoot 6 meters. So I actually took some home because I wanted to like study them a little bit further. Didn't know how well they shot. And they shot out of my Petri dish and landed on my wall. So this is how this thing spreads. They're really, really small though. So if you want to see these, you want to head out after things get really, really damp at Lake Merritt and start checking basically woody debris, and you'll be able to find these little kind of explosion looking things on there. And that is our cannonball fungus. We also have lots of sclerodermas at the lake. These are these things that kind of look like a puffball, but if you cut them open, they're discolored on the inside, or they'll be white starting to discolor. These things are actually kind of toxic to dogs. And so this is one of the ones that does cause problems for people that walk their dogs potentially at Lake Merritt. And these guys never really open up all the way. They'll get kind of old and they'll kind of crack a little bit, and the spores more or less kind of float their way out. We do have cup fungi at Lake Merritt. We get Pizizas in some of the wood chips in the gardens. And these can fruit in enormous numbers. The funnest thing about these is if you blow on them, they will spore disperse. And so all of a sudden, if you blow on it, and you like get your face away from it, it'll smoke all of a sudden. And those are all spores being shot out of all those asci at your face. And that's how it spreads its spores. We also have less mushroomy things, but they're cool anyway. I'm going to talk to you about them. Rust fungi. So these are things that actually attack plants at Lake Merritt. We have this one, it's the ground seal rust, which you can find on a lot of the different kind of weedy species of plants at Lake Merritt. And if you get up really close, they actually are really gorgeous looking. They look like little tiny blisters. We also have things that attack insects. You don't have to go to the tropics. You can just go to Lake Merritt. We've got tons of fly death fungus at Lake Merritt and in my apartment because I brought it home and infected the flies at my place. This is a fungus that attacks various things in Diptera, different types of flies. So at Lake Merritt now I've seen it on, I think, 2 or 3 different species of flies. It definitely goes after Drosophila, the 2 different Drosophilas you find at Lake Merritt, and I've seen it on a dung fly now at Lake Merritt. And it kind of makes them into zombies. So the spore basically gets on them, gets inside of them, and then tells them, your wings are broken and their wings can't work anymore. And that tells them, crawl up someplace high, and they crawl up really high. And then it says, bite into that substrate. So the fly will bite into the substrate. And then the fly basically dies. And this thing just rains spores all over the place onto other flies. So we do have pretty cool creepies in the fungal kingdom at Lake Merritt. We also have one of those that does that to spiders. And so we've got at least one tree at Lake Merritt that I can consistently find these on in April. And this is entomopathic fungi that goes just after spiders. And so you'll find these little tiny spider bodies. You can kind of see a leg sticking out of one of those photos there. And then these like fruiting bodies will come off of it and then rain spores down on the other spiders that are on that tree. They're relatively small though. So that bottom picture there where my finger is, inside that little tiny wedge in the bark, you can see a little round circle. That's the size of these guys. So really, really tiny. We also have other kind of fungi that attack insects at Lake Merritt, but these ones you'll usually find if you're turning over logs. You can find beetles a lot of time that will have this white muscardine fungus attacking them. I've seen this only a handful of times at Lake Merritt, but it is one of the most common fungal pathogens of insects in the world. You can pretty much find this in any habitat, so it's not too surprising that we can also find it at Lake Merritt. And my favorite one, because it doesn't kill the insect, but it does get to ride on the insect, are basically these ladybug STD fungi. These are transferred from ladybug to ladybug when they're doing their business, and they attach to the outside of the shell, and they make a living on the shell, but they don't actually impact fitness, surprisingly, of these ladybugs. And actually the ones in this photograph right here were picked up by a researcher in Harvard because he saw them on iNaturalist and said, hey, could you get me a collection of those? And I sent those to them. And those are being worked on right now in far as describing a new species, right? This is being used in a collection with a whole bunch of other ones, similar to being collected other places, but just goes to show there's still a lot of stuff out there that we don't know that's right under our nose in a place like Lake Merritt. You just got to get out there and start looking, because the hardest thing in all of this is just learning to develop your eye to be able to find things. And that's something that like a lot of us, if we've lived in the city all our lives, we're not really good at doing, which is taking time to look and find things. And so I'd say one of the best things you could do is go to Lake Merritt, to some place where there's a couple shrubs and bushes, pop yourself down and just stare, just zone out for 5 minutes. And you'll be amazed at what you start to see that you didn't see because you were busy moving around, usually Lake Merritt. There's a ton out there. That's basically a quick kind of view of the mushrooms that we've got. So with that, I'll open it up to questions, and I'll open up this chat window and we'll get back there.
Awesome. Thanks, Damon. We've been compiling a list of questions that have been running since you've started your presentation. Amy Patton wants to know, what's the weirdest or most interesting place that you found an urban mushroom?
Since I manage the building I live in, I have found Copernellus, unfortunately, in somebody's bathroom before, which, as somebody that is in charge of the health of the building, it is one of the last places you want to find that mushroom, because it means that some of your wood is being eaten.
Yeah, yikes. Yeah. Fanny F asks, phenolic smell, does that mean like old-time cough syrup?
Yes, actually a little bit of old-time cough syrup.
I think someone else maybe answered that in the chat window, and that it smells like old Band-Aids.
Yeah. Well, because they used to put phenol in some of the old Band-Aids as a way to kind of keep them sterile.
Right.
If you've also ever smelled any kind of like sanitizing solution, like an industrial sanitizing solution that janitors use, it also has a lot of those kind of phenol-like tones to it. So basically, if it smells like you might be cleaning with it, it's probably not something you'd ever want to put in your mouth. It's an easy, good life decision.
Nice. And Sheikah T asks, I'm curious, is there going to be a segment on psilocybins specifically?
I don't think there is one in here. Psilocybin is something that you do find commonly throughout downtown Oakland now. We at least have, I've seen, I think, 4 different species of psilocybin mushrooms in downtown Oakland. So they are there. It's just, they love landscaping wood chips.
I'll throw out there that that's also probably one of the most competitive mushrooms for urban foragers to collect, because of its popularity. It's up there with finding a nice king bolete or a big clean chanterelle. Let's see. Purnima says they would love to see a handout on edible mushrooms of the Lake Merritt region.
I can look into that, but like, once again, there's that giant caveat of urban mushrooms are picking up all kinds of funky stuff, especially when you talk about heavy metals. I can send you reams of paper of studies about this, which is why I would tread very lightly into deciding that you'd want to eat something from Lake Merritt. But if you want to learn to identify it and know that it's edible, I think that's something that I can work towards getting maybe a little PDF on.
Nice, nice. Eva wants to know, what's your favorite mushroom?
Oh man, it like changes like year to year. Like I know what, I think right now, like I still love like the fly death fungus, even though it's not technically like a mushroom, it's just so freaking cool to like have something that can take out your flies. And because I now am like the owner of an entire like multi-lineage of that fungus in my apartment.
You're a busy fella, Damon. Brett M has a heady question here for you. Do you have any recommendation for public databases of primers to use for sequencing of mushrooms? He works as a molecular biologist, big fan of Biorad, and has potential to do a few side projects and would love to have access to more molecular information.
So a good place to start is the University of Guelph. It has the Barcode of Life project, where they've kind of standardized a lot of barcoding primers. So they give you ITS primer sequences. And then from there, I would say that's usually where people start, is the ITS primers. And then for certain taxonomic groups, you might have to drop over to other primers. But that's where you have to kind of hit the literature and know the genera that you're dealing with. A great person to talk to about this type of stuff, and he has a couple of YouTube recordings on this, is local mycologist Alan Rockefeller. Does a lot of sequencing these days and is really into the bioinformatics side. And he's got YouTube videos already up on this. But if you need more information, my contact information was on here. You can shoot me an email. I can get you that information.
Great. Let's see. Haley wants to know, how have mushrooms been affected by this very dry fall or winter? Are all species affected equally? And I'll just say that's something we touched on a little bit earlier with Christian's talk, but it'd be interesting to hear your take on it as well.
So around Lake Merritt, we've got a really different location, right? Because there's artificial watering going on most of the year. And so we do see that some species, rain or shine, rain or no rain, it doesn't really seem to matter that much. So Agaricus xanthodermis, it could care less about what's going on, it seems like, at Lake Merritt. It's just going to fruit every September on clockwork. Other ones, though, which I'm kind of surprised by, is that some of the mycorrhizal fungi at Lake Merritt, so like Amanita muscaria, and actually I do find a couple of MycoBlitz in the area occasionally, those are very weather dependent. So those do need a big rainy winter in order to get enough energy to then shoot up fruiting bodies. So it really depends on the species.
Great. Sharon J says, a couple months ago, a patch of honey mushrooms were growing under her apricot tree. Will they start killing her fruit tree?
I've got bad news for you. They're already in there. When you're seeing the fruiting body, it means it's already been growing there for a while. And so what I might do is start to get emotionally detached from that apricot tree. And or you can do some kind of selective, maybe trimming of some of the dead wood, but most likely that honey mushroom has kind of penetrated the full tree at this point in time. One thing to think about, though, is when that tree decides to finally go, you might want to take a couple years off before putting another tree there, just to give a little bit of space for the mycelium to die down and any of the spore deposit to die down.
Solid advice. Yeah. Armillaria, never a good sign at the base of your favorite tree.
No.
Let's see. Daniel wants to know, are those entomopathic fungi present year round?
Let's see here. I find fly death fungus all year round. The spider one, Gabelia, or however one does not butcher the name, I usually find kind of January through April with. It seems like the high point on that one is usually April. So that one seems to like warm weather, but also when there's still some random kind of wet flurries from our weather system. God, please be that case this year again. So that's usually when you see those. It's more during the wet season.
I'm going to take an aside and use my liberty as a moderator to tell a story about you. Didn't you originally discover the fly death fungus in your apartment?
Well, I actually... The first time I ever saw it in my life was in Point Reyes, where you can collect mushrooms. And I was like, no one's really going to care if I collect this one. So I take it home, and I'm a home brewer. And so I'm a home brewer and a compost lover, which is never a good relation for you and your partner because you then share the place with flies. So I was like, you know what? I wonder if I could bring this home and if it would help. Like, I know they're different species, but what the heck? I'll just give it a shot. So I brought the big infected fly home that was a dung fly. I caught a bunch of the Drosophila from the house, put them in a jar, let them hang out just 24 hours, and then released everybody and was like, well, I guess we'll see what happened. And ever since that fateful April in 2014, I have a continual stream of dead flies with the fungus showing up, usually about 6 feet off the ground all throughout the apartment. And, at first I was like, hey, that's kind of fun. And then I started realizing, because the apartment manager, as I was cleaning the apartments and the hallways, I was starting to find them also in the hallways, 6 feet off the ground, dead, stuck to the walls. And I was like, oh, dude, what did I do? Oh, no.
Wow. Well, I mean, at least it wasn't, a wildly non-native fungus. It was a local species, and you just inoculated your apartment building. You're probably doing your tenants a favor.
I feel that I am. I'm protecting them from me, basically.
I feel like there's going to be a big rush of applications to live in your apartment building after today's program. That transitions pretty well into this question from Fanny. The way this was presented makes them think that each little area has its own population of fungi that would be endemic or landscaped by humans inadvertently. Am I thinking about this right?
You were thinking about that right. So we have a lot of endemic ones I think have made their way there because they were locally in our environment. And then the process of making a urban park gives them places that are wet all year round, maybe have the trees they like and things like that. And then we've got that whole other group that comes in with a lot of the landscaping techniques that people use in the Bay Area, which is wood chips and things like that, that then select for these kind of metropolitan fungi that you find in many places in the world.
All right. And I think just got a couple more questions here. Ryan wants to know, are there other factors besides rain that determine whether it's going to be a good year for chanterelles?
Oh, man.
If only, Ryan, if only we definitively knew that.
Yeah, Trent would not be here doing this right now. He'd be in the hills.
I'd be busy.
So I have a pet theory, and I'm going to throw it out there because it doesn't hurt me. You're not going to pick my patch from this knowledge. But I've noticed that when we have massed years of Quercus agrifolia, which basically means the years that coast live oak dumps a lot of acorns, I tend to see a lot of chanterelles in those years. So that being said, this is a great year to test that theory because how's the rain doing? Not too good. How's the mass season doing? There's a ton of acorns out there this year. So if this theory holds true, I should be seeing a decent amount of chanterelles once we get into maybe kind of later January and February, but we'll see. And other people can kind of watch that too. So you got two tree species that I watch for mass. I watch the Quercus agrifolia, the coast live oak, and I watch the California bay. They're both having mass years this year.
Interesting. I'll tag onto that with my own anecdotal information. First, I have to remind our viewers out there that unfortunately collecting fungi or any materials at all is prohibited in East Bay Regional Parks. That being said, I've noticed that the last 2 years have not been particularly abundant for chanterelles and other ectomycorrhizal species such as russulas, the boletes and swillus species, so on and so forth in the East Bay Hills. I feel like our really compressed rain cycle is sort of dramatically affecting those ectomycorrhizal species, which are species that are associated with a host tree, a big host tree. So that's interesting to hear your theory about it, Damon, because yeah, our coast live oaks and bay laurels are both having pretty abundant mast years right now. So I don't know. Yeah, stay tuned. We'll see what happens. Last question, and then we're going to transition into the iNaturalist MycoBlitz results. Eva wants to know, have you eaten any mushrooms from Lake Merritt?
Oh God, too many of them probably, which, bad on multiple levels. There's not obvious signs about collecting mushrooms at Lake Merritt, but general rule of thumb is not okay. And then second of all, urban habitat, gross, heavy metals, human things that end up on the ground at Lake Merritt. Not something I'd want to go into, but yeah, I've dabbled in a number of the species there.
Yeah, I'll follow that up, Eva, and say that I once collected a shaggy mane. It's a local edible mushroom from the side of a parking lot down in the South Bay and sauteed it up and ate it. And it was just about the worst tasting thing I've ever put into my mouth. It literally tasted like metal. So yeah, again, exercise caution and scrutiny when collecting edible fungi for the table from urban areas, especially near roads.
Yep.
Yeah.
And just to put it out there again, I mean, I think Lake Merritt's a great place to go see mushrooms and get your skills on how to see them, find them, identify them. But for eating them, for like forageable mushrooms, like I haul my butt up to Salt Point, to Point Reyes, or all the way up to Jackson Demo if there's things I want to put in my mouth. And that's A, from the legality side of it, and then B, also from my personal health side of it. I'm trying to accumulate the least amount of lead and mercury as I can during my short stay here in Oakland.
Oh, aren't we all? Well, Damon, thanks so much. That was a wealth of information on the local species of Lake Merritt, and thanks for answering all those questions. And yeah, if your throat's not too parched, I'm going to roll this directly into the review of our iNaturalist MycoBlitz.
Let's do it.
All right. Folks, our viewers out there, as many of you probably know, we normally hold this event in real life, or IRL, as they say on the internet. And we fill the nature center with tables covered with piles of mushrooms in various states of decay. And then we invite a few thousand people to come up here over the course of a weekend and poke at them and smell them and handle them and get to know them. Since that wasn't a possibility this year, what we did was we hosted a virtual collection event based on iNaturalist, in which local folks to the San Francisco Bay Area went out and digitally collected fungi by documenting them and uploading them to iNaturalist, as Damon so well explained during his talk. So what we're going to do now is share some of our finds from the week leading up to this event. So the collecting event officially started last Saturday and it ended this morning. So I'm going to screen share and take you over to iNaturalist right now. And here we are. Yeah, here's our project page, 2021 Tilden Fungus Fair MycoBlitz. First off, I just want to say I'm kind of blown away at how many observations got crammed in just the course of a week. 6,286, 497 species, and almost 1,000 people were out there documenting fungi in the Greater Bay Area, the 966 observers. That's a lot. And perhaps that's because I included a pretty big range for this event. I just used iNaturalist's sort of generic placeholder that was Greater San Francisco and Monterey Bay. But I did that because typically for this event, the Fungus Fair, I accept collections from as far north as the Jackson Demonstration Forest, or even farther and as far south as Santa Cruz and Monterey counties. So I wanted to encompass a good portion of that for this event. So that's what we did. So if you're on this page already, I'll drop this link into the chat in just a few minutes so you can check this out on your own. You can navigate, you can see recent observations. iNaturalist just has this fantastic interface now where you can sort by grid, you can put it on a map, or you can list them out. I really like the grid view because it gives you these sort of snapshots of people's mushroom photos of varying quality. It's always a challenge to identify fungi from photos alone. Like Damon mentioned, it's important to try and capture the salient features that will help you identify species. So, we generally want a shot from the top, a shot from the bottom showing the gills, pores, or fertile surface, and then a shot of the mushroom in its environment. And that's how you can increase the odds of your specimen getting identified on iNaturalist. So I'm not going to go through all 6,000 observations, but I am going to go over to the species observed here. And Damon, I was thinking you and I could just kind of talk about the top 5 that were observed here.
Okay, sure.
And before we tear into this, I feel like it's my duty to note that just because these were the most observed species doesn't mean they're the most common, right? So iNaturalist doesn't control for effort. And there's a reason that a giant red and white mushroom is #1 on the list. And that's because it's really easy to find and people get excited about finding it. So a lot of people are going to go out and document this species, right? So this is not a reflection of abundance, but it is a reflection of occurrence, right? Yeah. So there it is, Amanita muscaria, the mushroom, the myth, right? This species is probably surrounded by more controversy than almost any other one I know. Just mentioning it in an online mushroom group, yes, those are a thing, online mushroom groups. This is kind of sure to start some sort of battle of wills, right? Basically, there are 2 types of people in the world, people who think this mushroom is edible and people who think it's toxic, right? And anytime you bring this up in an online forum or even at a fungus fair, you're going to get people on both sides of that argument. So I feel obliged to tell a story about it. I think both are right. By itself, this is a mildly toxic mushroom. And I think you mentioned this, Damon, that this is not one that you want to consume for kicks. It does have toxins in it that can affect your liver and other parts of your body. And it's very weakly hallucinogenic. So just really not what your average thrill seeker wants to pop in their mouth. However, I have eaten it one time, and I was actually with David Aurora. So, when David Aurora feeds you a mushroom, you're kind of like, oh, well, this is probably going to be fine. I mean, it's David Aurora. So I was with a 4A that he was leading, and then we went back to a place and cooked up everything that we collected. And one of the ones that he decided to cook was Amanita muscaria. And he prepared it by parboiling it, which is you put it, cut it up and put it in a pot of boiling water. And then he poured the water out, and then he did that again. So he parboiled it and changed the water twice and then fried it up. And we ate it, and I experienced no ill effects. And amazingly, after being boiled twice, it was still rather crisp, almost like lightly cooked celery. So I was kind of surprised by that. However, I'm not advocating for the eating of this mushroom because it had relatively no flavor at all after being boiled to death. And again, there are so many edible fungi out there in the world that really no one should be pushed to eat things that are just on the far edges of edibility or something that you have to specially process to remove the toxins.
Yeah, I've had the same experience with consuming this one. I've done the parboil method. I'm super paranoid because I'm a biologist. And so I think I parboiled it like 3 times. And it still was that celery cooked crispness. And I found the trick was you really had to get it really, really browned up in a pan afterwards. And then you had kind of these nutty tones to it, but it wasn't like an overwhelming, like, oh my God, look at all the flavor in that mushroom. And that's also why you find like some of the local, myco chefs, like the Chad Hyatts of the world. What he likes to do with that one is he'll parboil it, dice it up, and then make it in a fungal ceviche. Because it does have the texture of fish. And then if you basically coat it with lime and chilies, it fits perfectly. And so there are different ways to work with it. But like Trent said, it's one of those ones that it depends upon the spectrum of comfort you feel with identifying, processing something, and then like Trent said, there's fights online all the times about, it's basically like the David Auroras of the world who are like, this should be one of the first mushrooms people eat because it's easier to identify. You just have to learn how to cook it. And then other people online that are like, oh God, no, if you just touch it, you're going to die. And that's kind of the spectrum that you're dealing with then.
Right. So
for me personally, I feel like it's one of those ones I got to really late because I also had that kind of like, I know there's bad stuff in it. I don't know if I'm comfortable with the processing and things like that. And so I don't think I consumed one of those until, 10 years into consuming wild mushrooms, maybe.
Right. So interesting species from an edibility perspective. So this is an ectomycorrhizal species, which means it's associated with a host tree in our area. It's commonly found with Monterey pine. As you can see on the map here that I'm sharing, this species, or this species concept as we currently understand it, has a cosmopolitan distribution. It can be found all over, native to almost the entire northern hemisphere of the planet. And as this map indicates, it has been introduced into the southern hemisphere, again, primarily on Monterey pine, which is just a local tree native to central coastal California. So muscaria followed Monterey pine when it was transplanted around the world. Monterey pine is a very common tree grown for lumber in other parts of the world, and Amanita muscaria went with it on its roots. And so we humans have introduced this fungus to remote corners of the world, particularly New Zealand. Look at all those occurrences down there. So there's also a lot of Monterey pine grown in New Zealand. So there you have it. That's Amanita muscaria, classic. I will also mention, I mentioned this has a rich and storied history. In just a moment, I'm going to drop a link into the chat window. It is an old New York Times op-ed cartoon, a little short animation from about 3 years ago that has maybe the best synopsis of the cultural history of Amanita muscaria that I've ever seen. You should do yourself a favor and watch it shortly. I'll throw that link in the chat window in just a moment. Hyphiloma fasciculare, second most commonly observed species. These things are they're just everywhere, This is a super common saprobic species. Meaning it's a generalist decomposer. So typically found on decaying hardwoods in large clusters. In my opinion, that's why it's number 2. It's this bright colored mushroom. It's out there all over the place. It's easy to spot. It's relatively easy to identify, and people found a lot of them.
Yeah, it's definitely one of those ones when you're walking through the forest, it like literally jumps out at you. It's hard to not see it, just because that color is so darn bright. They do fade in time, and so we do get a lot of haggard-looking ones on iNaturalist that don't have an identification on them. But then if you get a closer look at it, you realize that it is Hypholoma, so that color does fade in time. But when they are babies, it is just this neon green, yellow color, like things you don't expect nature to make. And that's why it draws so many eyes.
Yeah, and isn't this one of our local species that fluoresces under blacklight, or is that the other Hypholoma?
Yes, yep, it's this one. It's probably the strongest UV fluorescent mushroom in our area. And so if you ever want a kick and you find one of these in the forest, come back with a black light. And you can get these black lights really cheaply these days. If you get like a pet urine light, they sell them all over the place for like $10. Go out in the forest with that, you get to see scorpions and tons of sulfur tufts glowing everywhere. But just an enormous amount of UV fluorescence from this. And like I said earlier, we're not really sure why they do this. You know, there's speculation that maybe because a lot of insects kind of see in these spectrums, that maybe they're advertising themselves to insects with that UV fluorescence, but we're not really sure. In fact, I'm not even sure what the compound is in them that fluoresces. Me and Alan Rockefeller a couple of years ago tried to do an SDS page gel, which is a fancy biotechnology thing for separating proteins, to try and figure out if we could figure out what, you know, molecule this was in there that was causing the fluorescence, and whatever it was didn't hold up very well to the damaging effects of the gel. You could never isolate it.
Interesting. Yeah, again, folks, this is one that you can go out and find pretty much anywhere, super common. This is mildly toxic. It probably won't kill you if you accidentally eat it, but it will likely cause you to empty your gastrointestinal tract. So don't take this one home for the table, but do go out and enjoy it. Easy to find, one that fluoresces under a black light. Pretty cool fungus. Let's take a look at this next one. We'll just review these top 5 here, and we've got to get out to Omphalotus. So, folks, we may run just a bit over, but don't worry, trivia will be coming up shortly. We'll roll right into trivia in just a moment. So, Choleriza umbonata, the redwood rooter. I was surprised to learn that this is Christian's new favorite mushroom right now, which strikes me as odd because it's so common. It's such a common and relatively boring species in my opinion, but I guess if you are from outside of the redwood range, this thing is fantastic.
It's pretty cool.
It's relatively big. It's famous for this super long rooting stipe, hence the name redwood rooter. This stipe can extend, 12 or more inches down into the redwood duff, the substrate in which it grows. And it's kind of just an interesting species. I actually read something funny about this species when I was preparing some notes for this, and that is Tom Volk, well-known Midwestern mycologist, says this mushroom is a reliable indicator for redwood trees, which otherwise can be elusive and hard to spot for mycologists who spend all their time crawling around on the ground. So there we go, using the mushroom as an indicator to look up that you're in redwood habitat.
And likely the reason it's got that long root too is to get through all that duff to things they can actually kind of break down. And it is, if you've never found one before, go to the redwoods right now. They are out in good numbers. And it is one of the most exciting mushrooms you'll ever pick, just because you'll grab it and you're like, okay, I'm just going to pull this up. And you're like, no, and it just keeps coming, and like sometimes you do get that, good 2-footer and you're like, geez, where's this thing going to stop? Really cool mushroom, that response. And it's actually a pretty sturdy mushroom. That stem is pretty pliable, which you don't always see in a lot of mushrooms coming up in the redwood duff. A lot of them have like kind of brittle stems. This one's got a pretty kind of rock solid-y, flexible stem to it.
Nice. Now the redwood rooter. Go out and find that right now under redwood trees. Our 4th most commonly observed species for this event is Armillaria mellia, the honey mushroom. As far as I know, this actually represents a species complex or a group of mushrooms that are morphologically similar and very wide ranging. But if we look at the range map, it's going to reflect that. Yep, look at that. Same species all over East and Western North America, all over Western and sort of Central Europe, sprinkled throughout sort of mainland Russia. So yeah, anytime you see a distribution map that is this big, it's either actually a very widely distributed species like Schizophyllum commune, which has been shown to be the exact same species all over the world, or it means that we don't understand enough about the species to actually break it down into the individual species that we're probably seeing. So, there's a chance that we have a specific type of armillaria or honey mushroom here in California. There's another type in the eastern United States, and there's another type in Europe. But Melia is the old name from the European fungal taxonomy from hundreds of years ago that we haven't redefined it yet, as we know. So I don't know. I guess this is a good time to remind people that species names, especially for fungi, are sort of just concepts, and they represent our best knowledge to date. So for what we know about the species.
And that's just a good thing to know, especially for Western North America, where those names are changing with some degree of speed, is know a name, but maybe don't get too attached to knowing that specific name, but know that concept of that species. Because I can't tell you how many things have gone through 4 or 5 name changes just since I started looking at stuff, like since 2009. I'm just like, how do you keep up? Well, I'll use the common name to kind of help me bridge that species concept until we get the next, new scientific name. And there's probably still going to be at least a couple more decades of major reorganization going on in fungal taxonomy as more genomic work gets involved and applied to how these things are related.
Right. It's still a very exciting time to be a mycologist, specifically one who's interested in taxonomy. I just want to say one more thing about the honey mushroom before we look at our last species that we're going to review, and that is that in the eastern United States, this species is famous for producing foxfire, which is the bioluminescence of the mycelium of this species. So this is a parasitic species which invades living and recently deceased trees. In our area, it's very common on hardwood trees, at least this species concept is. And so in the east, you'll get this mycelial network growing up through these recently dead or still living trees. And the mycelium is so strongly bioluminescent that you can actually view it. You can see it on a very dark night. And that phenomenon is known as foxfire. As far as I know, that's not the case for armillaria in California, right?
No, some of ours do have bioluminescence, Trent. I'm not sure if it's specifically Mela, but I know at least Sydney up in Corvallis has cultured some. I believe Hart also in Castro Valley has cultured some that are showing bioluminescence in culture.
Oh, wow.
So ours do have it. And I do hear about people occasionally that where I grew up in Calaveras County, that when they were moving wood at night, they would see glow on like damaged stumps. So it is there, but it is poorly documented, which would be, this is another great place to be an amateur mycologist. There's all this stuff that would be great to get good documentation of, where you can say, yes, that is here, and this is the species that's doing it.
Well, that's news to me. I'm pretty excited about that. I'm fairly fascinated with bioluminescent fungi. And speaking of bioluminescent fungi, we'll round out our top 5 with this one, Omphalotus olivascens, the Western jack-o'-lantern. Damon, you took that picture, and I liked it so much, I made it the species profile photo for this. So this is I'd say this is our locally famous bioluminescent mushroom species. It's saprobic or potentially opportunistic parasite on hardwoods, particularly live oaks here in the East Bay. It is fruiting in abundance right now. And it actually has gills that are weakly to moderately bioluminescent, right?
Yep. And like I said in my talk earlier, I've seen ones that look exactly the same, like during the day. I'm like, okay, both these will glow. One's glowing really bright at night and one is just dead. So you do kind of want to see a whole bunch of them. I've been hiking in the hills out in Martinez before, where I could, some were bright enough, I could see them at a distance and be like, whoa, there's a green spot on that oak tree over there. I go over there, and it's a giant clump of Western jack-o'-lanterns. So these guys can get really, really bright occasionally. I think David Aurora in one of his books said that I think the way to see it was you would get a really big fruiting body, you would get yourself a peanut butter and jelly sandwich, and you would sit in a dark closet, start eating the peanut butter and jelly sandwich. And by the time you're done with your peanut butter and jelly sandwich, your eyes would adjust it and you could read a newspaper by it. I still have not found newspaper reading, lightable, Western jack-o'-lanterns yet, but they do get really bright, but you just have to go out and see a number of them. And so what I would do is find an area which you're seeing them a lot during the day, and then go back there at night and let your eyes adjust. And you'll have a good chance of seeing them kind of in situ glowing.
Mm. Yeah, well, future goals as a mushroom hunter, find a glowing Omphalotus out in the forest. I'll just throw out one more thing, and that is that some new or recently minted mushroom collectors, particularly people who are collecting for the table, often mistake this for the California chanterelle. And that is a painful mistake because this is a mild to moderately toxic species. Apparently can make you sick for a number of days. So get to know your fungi before you bring them home for the table. Don't take home Omphalotus thinking it's a chanterelle. If it glows when you get it home and you get it in a dark room, don't eat it. How about that for advice? All right, folks. So let's see here. I'm going to touch on one more thing. Bear with me. We're going to get to trivia shortly. And actually, I'm just going to go over here to the observers. And I just want to throw out that Arlene Devitt, I hope I'm saying your name right, and I hope you're watching, Arlene, because you were the #1 observer for the MycoBlitz, 222 observations and 86 species. And for that, you have won yourself an autographed copy of Mushrooms of the Redwood Coast by Christian Schwarz and Noah Siegel. I will contact you on iNaturalist for your information, and we'll get that book out to you. And Shui, you came in 2nd place. And I know a lot of the mushrooms that you observed, Shui, if you're watching, were right here in the Tilden nature area. So thank you for that. You've won yourself an Opinel mushroom collecting knife. Not to be used here in Tilden, but you know, you can use it anywhere you can legally collect fungi. So big shout out to our 1st and 2nd place observers in the MycoBlitz. And a big shout out to everyone who participated in this event. Again, we had over 990 people that either intentionally or accidentally observed fungi and got them aggregated into this project. So thanks for that, everyone. And a big thank you to Damon. We really appreciate you sharing your local knowledge on the mushrooms of Lake Merritt.
My pleasure. Get out there and look, everybody. It's a lot to see.
Yeah, a lot of stuff happening right now. It's an exciting time to be in mycology. So friends, I am going to queue up my co-host, Melissa, and she is going to take you into a round of Kahoot mushroom trivia. And we tried to make this challenging enough that it will hopefully be interesting for even the seasoned mycologists in the group. There will be a 1st and 2nd place prize for the 1st and 2nd place in Kahoot. And so, you know, get ready to compete. Stand by.
All right, I'm just getting everything set up right now to share my screen.
Here, Melissa, while you're working on that, I see a few more questions came in the Q&A box, so I'll just try to answer those. Mark says, you mentioned that taboo on collecting in East Bay Regional Parks. It's not taboo, Mark. It's actually the law. Well, what are the rules on collecting in Golden Gate National Recreation Area? Unfortunately, it's prohibited there too. Again, the nearest places to the Bay Area that you can legally collect mushrooms are Point Reyes National Seashore and Salt Point State Park, the latter of which is sort of famous for its choice edibles and also for its competitive collecting. Otherwise, national forests in general allow legal collecting usually up to a certain amount. So again, commercial collecting is either discouraged, prohibited, or you require a permit to. But you know, hobby collecting, like if you or I want to go out and collect some for the table, that's allowed in most national forests. Check before you go, though. Google's your friend. Just Google your national forest that you're headed to and see what the rules are on their collecting.
Trent, can you see the Kahoot right now? Are you able to see it?
Yep.
I just shared my screen.
Yep, I'm seeing it.
Okay.
Sure thing.
All right, I'm gonna go ahead and drop the link in the chat as well.
Okay, folks. So I'm gonna sign out for a little bit. Melissa's gonna lead you in this very challenging game of Kahoot fungal trivia.
All right, if you've never used Kahoot before, you can either do it from your phone, on a tablet, on another device. You can also do it on the same device that you have in front of you. Just kind of minimize your Zoom screen. And I'm going to give it a couple of minutes so that we have lots of folks playing. Okay, and once you join at the link kahoot.it, you're just going to put the game pin in. It'll take you to another link and you can put your name in and then enter. And so you'll be queued in almost like a waiting room and you'll see yourself on the screen as well. I'm going to drop the game pin into the chat.
Melissa, if I may, folks, if you're out there watching live on YouTube or Facebook, you're welcome to play also. Just remember that if you make it on the podium, we're going to need your contact information. So if you're on Facebook, you can direct message us at Tilden Nature Area. And if you're on YouTube, we'll work out a way to get your prizes to you. But you got to win.
All right, we'll get started in 1 minute. See lots of folks joining. So 1 minute. All right, I'll go ahead and start. True or false? Fungi are more closely related to animals than plants. Are fungi more closely related to animals than plants? Good job, y'all. It's true. Really fast. Oh, David in the lead right now. And also I just want to mention the way that these points are also calibrated, not just the correct answer, but how quickly you click the correct answer. So that's how the score is determined. All right, so fungi are more closely related to animals. Fungi and animals evolutionarily diverged 650 to 900 million years ago. Fungi and plants diverged over 1 billion years ago. How are fungi different from plants? How are they different from plants?
This is a multiple choice. You can select more than one. Quickly.
Fungi cell walls contain chitin, and fungi do not produce their own food. All right. Fungi and plants are quite different. So fungi have chitin in their cell walls instead of cellulose, and they can't produce their own sugars like plants do. All right, which of the following are not fungi? Molds, algae, yeast, or lichens? The answer is indeed algae. So algae are not fungi. Unlike molds, yeasts, and lichens, algae are a group of organisms that are autotrophic. So they produce their own food. All right, without fungi, we wouldn't have these foods. Nice, we wouldn't have bread, blue cheese, or beer. All right, so many foods are created with fungi. Beer, wine, breads, and some cheeses are created with fungi. Yogurt is actually created with bacteria. Some fungi and trees share nutrients, sugar, and water. This type of relationship is parasitic, mycophilic, mutualistic, or saprotrophic. Good, mutualistic. So mutualism is beneficial for both partners. Ectomycorrhizal fungi grow on tree roots and share nutrients and water with the tree, receiving sugars in exchange, a mutualistic relationship. Fungi decompose blank, which provides a crucial link in the carbon cycle. Rocks, mycelium, soil, or wood. Wood is indeed the correct answer. Yippee is swift. Fungi are the wood rotters of the world. Some fungi excel at breaking down lignin, Trent? Allowing trees to decay rapidly and creating forests as we know them. After sexual reproduction, mushrooms release these structures to propagate: rhizomorphs, spores, seeds, or hyphae. Correct answer is spores. Spores are how mushroom-producing fungi propagate. Spores contain part of the genetic material of the parent organism and sail away on the wind to colonize new substrates. All right, true or false? You can tell if a mushroom is poisonous by its color. The answer is false. So there is no correlation between color and toxicity. Unfortunately, to learn toxic fungi, you'll need to learn more than just the color. You shouldn't touch poisonous mushrooms, true or false? True or false? Correct, it is false. You shouldn't touch poisonous mushrooms. Fungal toxins aren't absorbed through the skin. Even highly toxic mushrooms are safe to handle, providing you can keep them out of your mouth. All right, this one is going to be a typed answer. So you have to use your keyboard or your QWERTY board on your cell phone or device. What genus of mushrooms contain amatoxins, excuse me, a deadly compound? So then you must type your answer. All right, lots of correct answers. Amanitas. All right, amanitas contain amatoxins. Amatoxins are a fungal metabolic byproduct that can inhibit cellular metabolism in some mammals, which means they can kill you. This is another multi-select question. Where can you legally collect mushrooms near the Bay Area? Your own backyard, Salt Point State Park, East Bay Regional Parks, or Golden Gate National Recreation Area? Excellent. Your own backyard and Salt Point State Park. It's hard being a mushroom collector in the Bay. While you can collect on private property, most regional, state, and national parks prohibit mushroom collecting. Salt Point State Park is a rare exception. What is the scientific name of this prized edible mushroom? Boletus edulis. All right, this one is a type answer again. What is the name of this choice edible mushroom? It sounds like we need to start capitalizing as well. So what is the name of this choice edible mushroom? Chanterelles, correct. What is the phenomenon in which organisms produce their own biological light? Fluorescence, bioluminescence, convalescence, or illuminance? Good, bioluminescence. Bioluminescence is biochemically produced light found in organisms from fireflies to fungi. Bioluminescence is mainly produced by a compound called luciferin. Which is our locally common bioluminescent mushroom found on decaying hard wood? Correct, Amphilotus olivascens. Some word spellings. All right, so this is the Western jack-o'-lantern mushroom. Has weakly bioluminescent gills that can be photographed in extreme dark. Also known as the jack-o'-lantern. All right, Eve or Ev. Looks like you're our number 1. Carol is our number 2, getting 10 out of 15. And then we have Chi Hyphy as well. All right. I'm going to pause my share and I'm going to stop my share.
All right. Thanks, Melissa. I'm gonna tap back in here and just want to congratulate our 3 podium winners. If you're playing from Zoom or if you're watching right now through the Zoom webinar, please message us, just the panelists, to make sure we have your contact information, because we're going to be sending you out some special prizes for your efforts. If you're playing from if you're watching from another platform, Facebook Live or YouTube, go ahead and message us. We're Tilden Nature Area, and let us know your contact information. If you were one of those 3 winners, we'll expect you to reply based on the honor system here. Oh, thanks. I see our messages are coming in already. Great. What is the prize? Well, let's see. The first place prize is going to be the same as for the iNaturalist MycoBlitz. It's going to be an autographed copy of Mushrooms of the Redwood Coast by Christian Schwarz and Noah Siegel. Second place prize is going to be an Opinel mushroom collecting knife. And third place prize is going to be something that I'm going to decide in the next few minutes. Stay tuned. So folks, we are now winding down the 2021 Tilden Fungus Fair. So I'm just going to do a little bit of housekeeping here, and we're going to start to wrap things up. Thanks everyone for your participation. This was an experiment for myself and Melissa, and I think all of our presenters today. Again, we didn't want to let the pandemic get in the way of us getting together and sharing our mutual love of fungi. So even though we can't be together in person, hey, look, we all got together on a computer and stared at a screen and poked around a bit and hopefully learned some things. If you enjoyed today's program, it was brought to you by the East Bay Regional Park District free of charge. And if you are pleased with the service you received, you could consider making a donation to the Regional Parks Foundation. They're our nonprofit supporting agency, and they support interpretive programs and help fund things for us. So I'm going to drop the link into the chat box right there. If you're feeling generous, shoot them a donation. It's tax deductible. Our mushroom photography today that we viewed during our breaks was provided by me and Anna Towers. You can find both of us on Instagram at the following links. I'll drop those into the chat box right now. You all can give me a follow too. My account's been kind of stagnant this year. I haven't really been too inspired to post a lot. Today's music, for the most part, were provided by copyright-free artists on YouTube, and I included their name and album information on each of those videos. And then I played some additional music from my friend Dan Kirkwood. He's a musician based out of Juneau, Alaska. If you liked that sort of folky song you heard, you can check him out at Bandcamp. There's the link to his album, Obi-Wan Coyote. If you are interested in learning more about Bay Area fungi, you should join a mushroom club. You're lucky in that if you live in the Bay Area, we have a wonderful diversity of mushroom clubs. We have 3 local ones to choose from and many more as you go out away from the Bay Area. So I'm going to drop links to the 3 closest ones to the Bay Area right here. The Mycological Society of San Francisco, the Fungus Federation of Santa Cruz, and the BAMS, Bay Area Mushroom Society. So those 3 links just got dropped into the chat window. For those of you that were watching earlier, I reviewed quite a few mushroom guidebooks, and as promised, I'm going to drop all of the links to those into the chat box right now. So that was kind of a long list of books, but there they all are in the order that I reviewed them. I did leave out the one published in 1900 because that's out of print. You'll have to go and scratch around at your local used bookstore to find that one. And that is all of the information I have to share with you today. I think I'm going to drop that link to our iNaturalist BioBlitz into the chat box one more time in case you missed it. It's a pretty fantastic platform. If you don't use iNaturalist already, consider signing up for an account. It's a really great way to learn fungi on your own. So you just go out with your smartphone, you download the app, you take pictures, and you upload them And they're real humans that will try to identify them for you, like me. I'm on iNaturalist all the time, and I try to identify local fungi. So you can follow that link to iNaturalist to see our project page and learn more. Let's see, folks, if you're watching from Facebook Live or YouTube, I'm going to let you know that a copy of this video, along with all the associated links that I'm talking about that you can't see right now, will be posted shortly within the next couple of days, as soon as the video gets processed and edited for captions and everything. So again, if you're not with us on the Zoom webinar, but you're watching live on one of the other platforms, just hold your horses and we'll get that information out to you as soon as we possibly can. And at this point, I will just say that I'm happy to answer any last questions that some of our panelists didn't get around to. I may not be able to answer them all, but I can certainly try. Yep, iNaturalist MycoBlitz link. I just posted it in there. And let's see, lots of complaints about the Kahoot. I apologize, folks. That was just, that's the platform that the district decided to pay for. So that's kind of what I got stuck with using. It is a bit frustrating, but congratulations on those of you who navigated its terrible GUI interface. And if no one else has any questions, I think I'm going to wind this. Oh, thank you, the Amanita muscaria link. I promised I would post that, and then I didn't yet. This is fantastic. For those of you that are still watching, still tuned in, I'm dropping the link in the chat box right now. It's an old New York Times op-ed piece that's actually a little animated cartoon about sort of the history of Amanita muscaria. Totally worth a watch. Very seasonal. Let's see, Kate has an unanswered question. She went on 2 hikes and found some shaggy manes, took 4 and ate 2 and threw 2 in your backyard. Have fruit trees, was that a bad idea? No, shaggy manes are a generalist saprobic mushroom. They're not a parasite. So with any luck, you'll get shaggy manes growing in your backyard, but they're just gonna be digesting your sort of old dead grass and leaf litter. They're not gonna kill your trees. Yeah, no worries about that. All right. And well, Kate, I don't know where you picked those, but Mount Tam, yeah, I think they're illegal. So again, mushroom collecting, it's hard to be a collector in the Bay Area. My agency, the East Bay Regional Park District, doesn't allow commercial or personal collecting. And most of our national parks and state parks don't. So Point Reyes, National Seashore, and Salt Point State Park are the closest places you can legally collect. And you can collect for edibility or research up there. And then again, head on up to any nearby national forest if you want just wide open space and in general unfettered collecting. Thanks again, folks. And I'm going to wind this down with, I think I'll wind it down with our intro video here. If you'll just pardon the webinar countdown and just enjoy a little bit of a slideshow with some music outro. Thanks so much for watching, everyone. Melissa, thanks so much for hosting this event with me.
Of course, definitely. It was fun.
Yeah, this was great. This was a big experiment for us. So we haven't done a live program like this of this length yet. Hopefully it went well. I'm going to say one more thing, and that is, if you're still watching, if you were registered on the Zoom webinar, you're going to receive a follow-up email that has a poll in it. And it would greatly benefit us if you could complete that poll. Your answers will be anonymous, and it's just some questions to help us understand what worked and what didn't work with the event and what you'd like to see in the future. So thanks everyone so much for tuning in. I'm going to screen share this video, and we are going to very slowly fade out here. Thanks for watching, folks, and have a good one.